important thermoregulation. Basically, the skin receptors, they detect the change in temperature and converts the stimulus into the impulse. Simple. Detect the change in temperature, and then they convert that stimulus into a nerve impulse, which is sent along the neuron. So now, detection is a marking point, and also conversion of the stimulus into a nerve impulse is another question, another mark. Explain why structure B dilates on hot day. Why do we have a big dilation here on hot day? Yeah, why do you see your blood vessels on hot day? Yeah, the same thing I was talking about on the skin. Eh? Guys, there is no way in this life science you're gonna fail this bone. There is no way. Yes, what you need to do is to do what I'm telling you. Download the distinction material book, use it, make sure that you get a distinction. I'll be happy where I will be, whether I know you or I don't know you, as long as you're happy. All right, let's continue. They're saying that we are saying that yes, here check how they are. They are big, ne? Yes, they are big. You see, much heat is lost, so it dilates so that it can supply, allow more blood to flow near the skin. You see, more blood is flowing near the skin. More blood is flowing near the skin, and as it flows near the skin, heat is lost. That's what they are trying to say. So allows more blood to flow near the skin so that more heat is lost. Yes, that's it. That's why it dilates during day, hot days. Sometimes we can also say that it dilates so that it can supply more blood to the sweat gland, so that the sweat gland can become more active. And once it becomes more active, more sweat is gonna be released. Yes. Next question. The next question is saying, what are the workers in some factories are constantly exposed to loud noise. Okay, this is just a story for a long period of time. This can destroy the hair. Okay, it can destroy the hair cells in the organ of Coti. They can destroy. Okay, we know that now the loud noise. People who are staying in towns where beep 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 all the time, beep beep. Hey, this one is making noise. This one is putting on music. Take note of that. Damage and the auditory nerve, resulting in hearing loss. Okay, survey was conducted in the developing countries. 2014 to 2018. To establish the number of factory workers who suffer from this hearing loss. Results are shown in the table below. Question number 1. Name the structure where the organ of Coti is located. Cochlea. We call it cochlea. Yes. Calculate the percentage increase. Yes, percentage increase. We like to ask you this. Ne? I don't know why. Maybe students, they don't get it right. Yes. Here percentage increase is equal to final minus initial over initial times 100%. Final, what is final? 2018. So my final is gonna be this 130,000 minus initial, which is 85,000. Yes, divide by the initial 85,000 times 100%. So you calculate using your calculators. What is the answer? Around maybe 52.9%. Yeah, so that becomes the percentage increase. Sometimes they can say percentage decrease, same thing, but that might be Yes, it must be maybe in negatives. Anyway, let's go. They're saying that suggest one reason why increase in the number of factory workers with hearing loss caused by exposure to loud noise in this can suggest one reason for the increase in the number of factory workers with hearing loss caused by exposure to loud noise in this country. They said it here. They said it here that this can destroy the hair cells in the organ of Corti and damage the nerve to the nerves. Yes, they said it here. That's why I like the passage. You get the answers from there. Draw a bar graph. Bar graph, you want me to draw? I don't think so. But what is important here is you have to be with a caption with the two variables. Yes, meaning that you have the variable of the year and the number of workers with that problem. And then we use the ID method. ID method. You remember the ID method? You remember the ID method? Some of you don't know the ID method. Let me show you the ID method. All right. The ID method is, you say, ID X Y. Independent variable on x-axis, dependent variable on y-axis. This is the independent variable, and then this is the dependent variable. Simple. And then now it means that number of workers is gonna be on the where y-axis here, and then number of years is gonna be where here. Simple. And then if you look at this, because there is increase, you will find out that it will have that shape. Don't forget, how do we mark? This must be years, but years, yes. If it has units, make sure that you put units. If there is no units, the same thing. And then here numbers, you must put the units. If it's there, make sure that the distance between here and here and here and they are the same. Make sure that these bars, they have the same breadth, meaning that how big they are must be the same, or otherwise you're gonna lose marks. Yes, so we mark the caption, we mark the correct type of the graph, because some people, they will draw a histogram instead of a bar graph. We mark the plotting, we mark the axis. Yes, we mark the axis, x and y, and then also mark the scale. Scale, make sure that if you start 0, 2, 4, for in this case, it's gonna be what? It can't start from 0. Yeah, why? Because it's not that big. Maybe you're gonna start from 5,000, 50,000, 50,000, 100,000, something like that. Or you will see the best scale you can use. Yes. So that is the question. Hope I'm clear there. Let's go to the next question. Which question is this? Question 3. Meaning that now we have 100 marks. Mahala. 100 marks. Yes. Meaning that 100 out of 150. How much is that? Yes. So now you know how much you have. Yeah, tell me, is the paper easy, difficult? Hey, how about it? Tell me something. Was the paper easy? Or is this paper easy? Yes. If you find it again, is it easy? That is paper for 2023 final exam. Yes. People tried to write it. All right. Let's go to the next question. They are saying that the graph below shows the concentration of abscisic acid in the cherry tree during the different seasons of the year. They are saying that this tree species loses all the leaves in the autumn and goes into state of dormancy, meaning that this tree, in science, we call them deciduous plants. Plants which lose their leaves during the period of what? During the period of winter. Yes, we call them deciduous plants. Yes, question 1. Question 1. They're saying that I need to be a little bit faster so that I finish. The first question, they're saying that during which month was abscisic acid concentration lowest? It's obvious, guys. During which month? Check this is concentration. November. Simple. November. Explain the trend of the graph from March to May. They're saying explain, not describe. Why? From March, they said March, from March to May. When they say explain, you have to give a reason. Yes. From March to May, there is an increase in the concentration of abscisic acid. Why? Because, don't forget, as they say that abscisic concentration, different season of there. This tree lose all the leaves during autumn, meaning that the abscisic acid is the one which is important for shading of the leaves. So it means that why does it increase? So that it triggers that shading of the leaves, so that the plant can undergo dormancy. So you mentioning increases, yes, a mark, and also explaining why does it increase, so that it can bring about the shading of the leaves during this period, so that it enters when it is already dormant. Does not lose a lot of water during winter, because it doesn't have that water in the system. All right, state one reason why dormancy in cherry trees. Suggest one reason why dormancy in cherry trees during winter month. Why do they shed? Why do they undergo dormancy? So that it minimizes the amount of water loss to the atmosphere. Yes, don't forget that water loss is most especially through leaves. So when you shed the leaves, it means that you are minimizing the amount of water being lost to the atmosphere during this period of dryness. Yes, the next question. Geotropism. Geotropism. Yeah, how about geotropism? If you look at geotropism, what are they looking at here? Geotropism is a plant response to the gravity. Sometimes it's called gravitropism. Movement of the part plant response to gravity. Oh, they have talked about this. Tropism is controlled by auxins. Describe the role of auxins in the roots. Basically, auxins are found at the tip of the root. And auxins, they bring about growth. So the first role is it brings about growth. But how does it bring about growth? High concentration of auxin will inhibit that part which has high concentration of auxin will inhibit growth. So basically, that's what you talk about, meaning that the part which has low concentration of auxin will grow faster, and then to bring about the bending action of the root. Then they're saying that 3.22, when a plant is placed horizontally, okay, you place it horizontally with light coming from all the direction, auxins will accumulate on the lower side of both stem and root. Explain the difference in a response on the root. And okay, here they are asking you that if you have light, you place the two things horizontally. Let me show you here in a different what here. They are saying that okay, you have a plant, you place here and you also place here. If this is a shoot, and then this is a root, yes. But if you have light from top, yes, from top, what's gonna happen? You're gonna have high concentration of auxin here. Don't forget that the role of auxin in roots, it is brings about growth. Even here brings about growth, but in roots, high concentration brings about inhibit, and then high concentration in root brings about promote. It means that this side is gonna grow faster, bringing about bending upwards, while this one is gonna grow slower, and this one is gonna grow faster, bringing about the bending activity. Yes. So basically, in roots, the leaf, the shoot will bend towards the source of light, whereby the high concentration of auxins will stimulate that growth. And then in the roots, the root is going to bend away from the light, meaning that high concentration of auxins in a root will bring about inhibiting. So this is what I'm saying of the inotropism. If you look at our distinction material book, yes, that's what they are saying. High concentration of auxin bring about that bending towards, you see. And then in the roots, high concentration, check the high concentration is here. It brings, you see, high concentration is here. It brings about the bending. The one which has no high concentration bends, grows faster than the one which has. That's why you see these ones that are too small, while these ones are longer. Don't forget that growth is elongation of the cell. Yes. So basically the upper part will go faster than why? Because the auxins will be high at a lower side, meaning that the upper side will grow faster than the lower side. So that's what they are talking about there. The second question is, so here what they are looking for is to show us that in roots, high concentration promotes, resulting in the bending of the shoot to the source of light. And in the roots, high concentration result in inhibition or inhibiting or limiting growth, resulting in the root bending to the ground or away from the light. Yes, let's go to the next question. The next question is talking about, what is talking about? Yes, independent variable and a dependent variable. Here, I'm not going to waste too much time because we always ask you questions concerning about this. We don't bring the same question, but I'm seeing that my battery is dying. All right, let me just say, name the gland that secretes aldosterone. Let's just go straight to the question without, you have to read here. But what is most important here is this. Scientists investigated the influence of decrease in aldosterone level on the blood. Yes, scientists investigated, yes, aldosterone level in the blood. So this is the most important thing. Yes. So if that is the most important thing, so it means that it can help us to know which one is independent, which one is dependent. Identify independent variable. Aldosterone is secreted by adrenal gland. Adrenal gland. Yes. And then independent variable, they are saying that investigated the influence of increase in aldosterone level on the blood pressure. Blood pressure versus influence of increased aldosterone level. This versus this. So it means that blood pressure is being influenced by this. So blood pressure becomes dependent, and then the influence of aldosterone level is dependent. Then the next question is saying, give 2 reasons why results may be considered reliable. The reliability, don't forget that you repeat the experiment. Did they repeat the experiment? Yes. Let's check. Yes, they said that they repeated the experiment. They measured every, this is average. Ne? Is it average? No. They measured every 4 hours. Every 4 hours for 12 hours. Meaning that they kept on repeating. And then they said that they calculate the average pressure. That is average. So meaning that they calculated for average, and also they repeated over a period of 4 hours for 12 4 hours for 4 hours for every 4 hours, for every hour for 12 hours. They repeated it 12 hours, 12 times basically. Yes. They're saying that explain 2 reasons why there was important for participant for the same diet. Yeah, so that you have the same maybe salt concentration, and then also to keep the same blood pressure, because if you have different types of food, it will bring, because they said that it brings about blood pressure. Yes, somewhere there. So I'm just rushing so that I go to the last question, because that is an open question. This is not that we're gonna bring the same question, ne? Yes, so it's just how do you extract it from the question. They're saying explain why participant blood pressure was measured before the start of the investigation. Whenever you do that, it means that you are looking for the reference point, basically to act as a control, or to have a reference point where you know that, okay, without it, or without it, what will happen, or before you applied the experimental measures, what was the conditions before, so that now you can bring about comparison. So basically is to compare the results. Or reference point. Explain why the level of salt in the urine of the participant expected to decrease after being injected with the aldosterone. Don't forget, the function of aldosterone is to stimulate the nephrons so that there is more reabsorption of the salt back to blood. It means that you have less salts in the urine, and more will be reabsorbed back to the body. Yes, think I'm clear there? And then the next question, yes, is the last question, I think. That is the eye. I know you like the eye, ne? You like the eye the way I like it. Yes, I think you like the eye the way I like it. So they're saying that what is A? A is vitreous humor. B is yellow spot. C is blind spot. D is lens. E is cornea. F is iris. So you have to know them. Identify F, we have said it. F, it is iris. And then you're saying state two functions of A. Where is A? Function of A. It brings about the shape of the eyeball. Yes, it brings about, ne? Yes, it maintains the shape of the eyeball. I stated it here in the distinction material book very, 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 very well. If you have the eye, then it means that you have this book, as I told you, that there is no way you're gonna fail. Yes, check it here. I said it here. This is aqueous humor. Change, yes, changes the shape of the near and far distance. No, that's the lens. That's not is here. It gives the shape of the eye. It gives the shape of to the eye, meaning that it brings about that shape of the eyeball. Then number two, state the function of A. Okay, we have seen it. Describe the structural difference between B and C. Structural difference between B and C. B is yellow spot. That's where the clearest image is formed. So it has more rods and cones. Highest concentration of rods and cones are found in B. They are saying structure. And then C has no photoreceptors, has no rods and cones at all, meaning that if the image forms there, you'll be blind. You won't be able to see that object. That's why you're saying that it is the blind spot. The next question is saying that describe this. Okay, name the visual defect occurs when the curvature of the eye is uneven. When the curvature of the eye is uneven, we call it the curvature of the eye, ne, is uneven. So we call it astigmatism. So we said it here, astigmatism is when the cornea or the lens is not evenly rounded in all directions. It means that it's not evenly distributed. So it is called astigmatism. Then they are saying that explain why sight on person, explain how the sight of the person will be affected if cataracts develops. Remember, we said that cataracts here is when the eye is cloudy, the lens is cloudy. So it means that light will not be able to go through. The lens becomes opaque, you see. So it means that you will be having this blurry vision. Yes, that's what they are asking you. What will happen? You will have blurry vision because light rays will not go through the lens. Why? Because the lens has become opaque. Check. Use distinction material. There is no way you're gonna fail. Yes. The next question, they are saying that describe the process of accommodation that takes place when the object is less than 6 meters. Buffet. Still, buffet. This paper, buffet. Ne? It is less than 6 mirrors. This is more than 6 mirrors. So memorize one and then do the rest. Just have to memorize one. We are saying that ciliary muscles contract, suspensor ligament slacken, tension on the lens decrease, the lens becomes more convex, light rays is reflected to the retina, or more light rays are bent more, and then a clear image is focused onto the retina. I'm rushing because of the electricity is gone. However, my system is dying in the next 3 minutes, yet the electricity is coming back within 7 minutes. So it means that I will be having that time when I don't have electricity. So you guys, I'm going to say that thank you very much for being here with me. Yes, I was discussing the final paper for 2023 Life Science. You saw that this paper was easy. Yes, this paper was really easy. Most especially those people who used my distinction material. Yes, those people who used my distinction material, I know that you got this paper right. Ne? Yes, you got this paper right. Thank you very much. I think I'll say that may God bless you. Don't forget to subscribe, like, share to others for our daily new video. Paper 2 is coming. Ne? Paper 2 is coming immediately when they write. Ne? After writing, I'll do the memorandum for you so that you test yourself. Thank you very much. Love you. We're going to be looking at this paper, the final paper for 2023 Life Science. Let's start with the first question. That is section A. So you have to download that paper from ThunderEDUC.com. Yes, if you want it, yes, you can find it on ThunderEDUC.com. Yes. All right. The first question, I always advise students that you cannot go to the paper and you leave objective unanswered. Even if you don't know the objective, at least try to guess. And then you never know, your guessing could be correct. And then you get marks concerning about that objective. So you cannot leave it because you don't know it and then you leave it unanswered. So you must always try your best so that you can get a mark, at least those 2 marks for that question. So let's start with the first question. It's about diabetes. They're saying that diabetes mellitus, as I usually tell you that from our distinction material, that when you talk about diabetes, you have to talk about mellitus. Don't just say diabetes. Diabetes mellitus or diabetes insipidus. Diabetes of sugar and diabetes of water. So this is diabetes of sugar. So it's caused by number 1, they are saying that over secretion of glucagon, no, under secretion of glucagon, then over secretion of insulin, and then under secretion of insulin. Yes, the best answer is the under secretion of insulin, because this diabetes is what you usually call sugar diabetes, meaning that the excess glucose cannot be converted to glycogen by the liver. Yes, so it means that you have too much sugar in the body, and then you end up suffering from this condition called diabetes mellitus. So the answer is D. So you expected to have 2 marks there. Then let's go to the second question, saying that receptors that are stimulated by the low water level in the blood are located in the where do we find these receptors which is stimulated as a result of low level of water in the blood or in the body? So which receptors are those? We call them, because we are talking about water, so it means that is osmo. So it means that those receptors are called osmoreceptors. Yes, sometimes we call it they are found in the thirst centers in the hypothalamus. So in this case, the answer is going to be B, hypothalamus. Don't forget that it is hypothalamus which determine which produce ADH. Yes. So this receptor are found in the hypothalamus. 2 marks there. They're saying that myelin sheath on a neuron on the nerve cell. What's the function? Provide electrical insulation. Yes. Transport impulse towards the cell body. That is a dendrite. Receives impulse from the axon. That is the effector. Converts converts stimuli into a nerve impulse, and that is a receptor. So this is a receptor. Basically, this is a receptor, this is a effector, and then transport impulse towards the cell body. This is the dendrite. So the answer becomes A. So expect that to be A. Sorry for that. And then we are saying that so the answer is M. They're saying that the structure where sperms are temporarily stored in the testes, the structure where the sperms are temporarily stored is the, how do you call that structure? Yes, definitely, yes, is found in the testes, but not all the parts of the testes will store the sperms. Hence, we will not take testes. Epididymis, that's where when sperms are being produced in the seminiferous tubes, yes, they are not yet mature to full capacity. So what does it do? These sperms, they go to the epididymis and they stay there for a short period of time so that they can mature fully, and then they can be used for fertilization. So the answer becomes B. D, this is the vas deferens. The vas deferens is just a tube which transports sperms from epididymis to ejaculatory ducts, penis. Obviously, everyone knows the penis, even if we don't explain it. Yes. Then we go to the next question. The next question is talking about which one of the following is a function of amniotic fluid. What's the function? Many students, I've seen that many students talk about amniotic fluid. They relate it to nutrition because it is a fluid. You expect it to be nourishing the fetus. No. Amniotic fluid is not related to nutrition. That's why even here on the objective, the first answer they gave is provide nutrition, so that it attracts the attention of the people who don't know the actual answer of the amniotic fluid. Therefore, in this case, the answer is going to be, it's not going to be nutrition, so this one is out. Yes. Then protects the fetus against mechanical injury. This is correct. Let's see if there is a more correct answer. Supplies oxygen to the fetus. No. And then removes metabolic wastes from the fetus. No, that's not a function of amniotic fluid. So the actual answer is going to be B. So there you get 2 marks. I hope yes. So let's go to the next question. The brain. Yeah, they're talking about the brain here. Brain is simple. I always tell my students that before you answer any question, you see this 2023 final paper. Yes, I expected everyone to get all the marks. Yes, to get all these marks. Yes. But you know, as a paper, as a past paper is always easy, but a paper where you go and sit, it is always difficult. But you can see it here as an easy paper, but when they tell you to sit on in exam room, and then we give you when you don't know, it becomes difficult for you. But as long as you prepare, this passing is a little bit simple. Yes, is a little bit what? Simple, as long as you read. Make sure that you remember what you have read by repeating and repeating and repeating what you have read. All right, number one. What is number one? Is corpus callosum. Number two. Number one is corpus callosum. Number two is cerebrum. Number three is cerebellum. Number four is the medulla oblongata. Don't forget that. I always tell my students that this first swelling is not medulla oblongata. It's called a pons, but we don't usually ask it. The second swelling is what called a medulla oblongata. You must always know the functions of each. All right. Which one of the following represents corpus callosum? Yeah, obviously we have said that one represents corpus callosum. So A is going to be the most correct answer in that regard. Then they are saying that the 1.7 is saying which one of the following is a function of part 3. Part 3 is what? Yes, we have seen that. Part 3 is the cerebellum. Yes, cerebellum is responsible for coordination of voluntary movement. There is a confusion. I always tell my students that don't confuse coordination and control. It does not control voluntary movements. It coordinates voluntary movements. Yes. All right, let's go to the objectives. They said control voluntary movement. No. Doesn't control. You see, they always give you a confusing answer as A. So make sure that you don't rush when you're answering such questions. So it coordinates voluntary movement. So the answer becomes C. So that is the most correct answer. Controls voluntary involuntary actions. That's not the function of cerebellum. That's the function of medulla oblongata. Controls all sensation. Receives and interprets all sensations is done by number 2. So the actual answer here becomes C. Coordinates all voluntary movements. Placenta is formed by. There is a video which I created, and it was showing you the animation of the placenta and how does it form. So here is number 1. They are saying that amniotic fluid and amnion. Amniotic fluid, no. Chorionic villi and endometrium. Yes, it could be the answer, but let's see if we have the most correct answer. Always read all the objectives and find out the most correct answer. They're saying that amnion, no. The moment there is amnion, no. Amniotic fluid and no. Don't forget that during the last stage of the development of the zygote until implantation, we say that it forms a hollow ball of cells, which we call the blastula or blastocyte. So this blastula forms the outer layer, which we call the chorionic, which we call the chorion. The chorion forms finger-like projection called the chorionic villi, which are used for implantation. And implantation occurs on the walls of the endometrium or the inner lining of the uterus, which we call the endometrium. Hence, endometrium and the chorionic villi becomes the part of this, forms what you call a placenta. Hence, B becomes the most correct answer. So, let's go to the next question. Oversecretion of hormone produced by thyroid gland may result in a person oversecretion of a hormone produced by the thyroid gland. Which hormone is that? First ask yourself, which hormone is that? Thyroid gland obviously cannot produce luteinizing hormone. Definitely is going to produce thyroxine. Thyroid gland, thyroxine. Thyroid gland, thyroxine. So you have to know that. Yes. So meaning that they're asking the effect of thyroxine when it is over secreted. Don't forget that you have what you call under secretion and over secretion. So in this case is over secretion. I stated it in my book for distinction material very, very, very well. And actually the way they brought it here is exactly how I try to explain it. I'll show you. Let's first find out the answer. Gaining weight because of increase in the metabolic reaction. No. If there is increase in the metabolic reaction, we expect to have low weight. Don't forget that thyroxine is related to metabolic rate. High production of thyroxine, you expect to have a high metabolic rate. And if you have high metabolic rate, it means that the food is being broken down very, very, very fast. Meaning that you might not have food to be stored and then have this gaining. So in actual sense, it looks to be oversecretion of thyroxine will lead result in a person. So we have said that here is going to be weight loss. Weight loss because of increase in the metabolic rate. So we said that I'm going to show you on our distinction material book. There you'll find it in the at the end. Yes, you'll find it here. Yes. Thyroxine is here. Yes, disorder which we call the hyperthyroidism. So here is the question, the answer of the question. They are saying that overactive of the thyroid gland occurs when the thyroid gland produces too much of the hormone thyroxine. Hyperthyroidism can accelerate the body's metabolism, meaning that metabolic rate is going to increase, causing weight loss, you see, and rapid or irregular heartbeat. Yes, this is what they are saying on this question, that oversecretion of this hormone will, which is produced by the thyroid gland, which we have said that is thyroxine. So it's going to cause weight loss because of the increased metabolic rate. That's what I tried to wrote here. That's where they got that question from. Yes. So I think I'm clear there. Let's go to the next question. They're saying the table below shows the average testosterone level of male of different age groups. Which one of the following is an explanation of explanation for difference in testosterone level between the age groups? Yeah, if you look at this, you'll find out that this is as a result of low level of testosterone. And what will make the low level of testosterone is either the person is still young, still low in age, meaning that not yet at that age, and then number two, the person maybe is having some infections. All right, let's look at this. Testosterone level are high in which one of the following is an explanation for difference in testosterone level between the age groups. 0 to 10 is 5, 10 to 5, 11 to 15 is 15, and then 16 to 20 is 19. So here they are saying group 1 and group 3 due to group 1 and group 3 due to start of puberty. No. Group 1 and group 3 due to start of puberty. Okay. Let's look at it. And then they are saying that group 1 than group 3. Does level of testosterone too much in group 1 than group 3? Check here. No. So this is out. Group 2, yes, than group 1. Yes, group 2 than group 1 due to the start of puberty. This one is correct, but we need to first find out if there is the most correct answer. Group 3 than group 1 because it needs to be inhibited. No. Group 1 than group 2 because it needs to inhibit. No. So basically the answer becomes 2. Why? Because group 2 is more than group 1 because group 2 has started is just entering the puberty stage. Therefore, the sperms are supposed to be produced in large amounts. Then let's go to the second question, whereby everyone must get all these questions. All these questions must be got right. Okay, number 1. The first question, they are saying that the process of maintaining a constant internal environment. How do you call that? We call it homeostasis. That's number 1. Good. They say organelle found in large quantities in the neck region of the sperm. How do you call those organelles? How do you call mitochondria? Yes, they're saying organelles. Yes, not organelle. They're saying disease characterized by degeneration of the brain tissue leading to memory loss. We call it degeneration of the myelin, sheath. Is called multiple sclerosis. But now here they're saying degeneration of the brain tissue leading to memory loss. Hence it is Alzheimer's disease. The layer in the eye that is richly supplied with blood vessels. How do you call that layer? If you look at it, is even dark in color, black when we are drawing it in most cases. We call it choroid, choroid. Yes, they're saying that the type of development in birds in which the young one is born fully developed and able to move, meaning that born prepared. Pre means prepared. Precocial. Yes, precocial. Then they are saying that the structure in the sperm that contains enzymes to dissolve the outer layer of the ovum. How do you call that structure which contains the enzyme? Acrosome. And then they're saying the blood vessel that transports blood, which is deoxygenated, meaning that high level of carbon dioxide from the fetus. From the moment they say from the fetus, where the heart is, remember, heart is, to the placenta, meaning that away from the heart. Any blood vessel which transports blood away from the heart is called artery. Artery. But because we are talking about the fetus, meaning that it's going to be umbilical. So it becomes umbilical. Yes, umbilical artery. You see? Yeah, simple. So you have got all those marks without even wasting a lot of time. Then let's go to the next question. A only, B only, A and B or none? A only, B only, A and B, both A and B or none? None. So you guys, if you don't write it the way they told you here, yes, the way they told you here, then you are just making your answers to be not correct, and is supposed to be debated about. So always do the way the instruction is telling you to do. All right, let's start. An extra embryonic membrane found in amniotic fluid. Amniotic egg, not amniotic fluid, sorry. How do we call that? Let me know if you have a question. You prepare. Now you know that paper 1 did not come out the way I expected it. So what should I do with the paper 2? I have to prepare more and more. Yes. So when you sit there in your rooms and then you relax, you think that you still have today and tomorrow, I'll read tomorrow. So when you watch this video, it will tell you that, no, I'm supposed to read more. I'm supposed to read more. 2023 must be my year. 2023 must be my year. So watch it, know your mistake. After that, go back and read paper 2. Be prepared for paper 2. I know that you will make it. Watch the videos on ThunderEDUC. You will never go astray. You will never. You will get a distinction. All right, let's continue. It's saying that. So here the answer is both A and B. Is both A and B. So a structure in the ear that absorbs excess pressure. As I'm always teaching you that they like this structure. I don't know why. Why do they like this structure which absorbs excess pressure from the inner ear? And then we say that that structure, it absorbs excess pressure from the inner ear, is called round window. It's not here. So it means that none. Then we go to the structure that transports semen out of the body. Out of the body is a penis. Yeah, that's where those semens are passing, but there is a tube inside the penis which we call urethra. So the answer becomes B only. Yes. Let's go to the next question. I remember I'd gone somewhere for specific research, and then I found the students. I want, whenever I go to any school or what, I want to be in the class. Why? Because teaching is nice. Teaching is nice. When you see that you are changing people's life, it's nice. Yeah, people's life can be changed because of you. Yes. So now I told them this, exactly this. I don't know whether those people have got it right. I hope. All right, let's go. Nervous system is either nervous system is either central. If it's not central, is peripheral. Meaning that A is the peripheral nervous system. Yes. So nervous system, the central nervous system is divided into two, either brain or spinal cord. Meaning that B is the spinal cord. Yes. The peripheral nervous system is either made up of cranial nerves or spinal nerves. Spinal nerves. So meaning that is spinal nerves. The spinal nerves, yes, these two, they form what you call autonomic nervous system. Autonomic nervous system. Autonomic nervous system divides into two, either sympathetic nervous system or parasympathetic nervous system. So now it's covered. Let's go to the questions and find out. All right, let's continue and find out. They're saying that identify the components of the nervous system represented above. A, we have said it. A is the peripheral nervous system. Done. And then they're saying that B. What is B? Where is B? B is the spinal cord. Done. Got it there right. Name the type of nerve found in C. C. Where is C now? Is remember, is not the nerves which come from the brain or the nerves which come from the spinal cord. That's why you call it spinal nerves. Cool. Done. Give the letter and the name of the component that slows down the heart rate when the emergent situation has passed. Meaning that sympathetic nervous system rises the situation, parasympathetic reduces the situation. I've been talking about this in the distinction material. Let me show you. Let me show you from the distinction material. Yes, even in paper 2, use this distinction material. There is no way you can fail this. This is nothing. This is nothing when it comes to our distinction material book. Let me show you. This is let me show you this. Yeah, it's here. It's not around here. Yes. Is here. Sympathetic nervous system, yes, is responsible for flight or you either fight or flight function. They're saying that the nervous system restores. Check. This one brings the situation to emergence, while Yes, this one restores it down when the situation is gone. Restores the body to normal state after the emergent situation. What are they saying here? Let's see. What are they saying there? They're saying that give the letter name of the component that slows down the heart rate when the emergent situation is passed. That's what they are looking for. Yes. So now I come here and they say parasympathetic nervous system. So the answer is E, and then name is parasympathetic nervous system. So this, what I'm explaining here is here. Let me show you. This diagram, that diagram is here. Is there? Simple. That's what they're asking. They brought this diagram exactly how you check. Yes, nervous system. Yeah, nervous system, central nervous system. Check. Central nervous system, brain, spinal cord. Brain, spinal cord. Peripheral nervous system. Yeah, where you have the autonomic nervous system and the somatic. Yes, and then now autonomic nervous system is divided. Everything is there. So there is no way how you can fail when you are using this distinction material. Download it from the link below. You will be able to get the distinction material book. All right, let's continue. They're saying that name the neuron, name the nerve cell that is made up of the nerve tissue. How do you call that nerve cell? How do you call that nerve cell? Is called a what? A neuron. Neuron. Yes. So we call it a neuron. Don't forget that a nerve, when you say nerve, it's like a wire. You know, a wire, solido, the wire which transmits electricity. But if you check inside, so this one becomes a nerve. When you check inside, it has small wire. Those small wires, we call them neurons. We call them what? Neurons. That's what they're asking for. Yes. So nerve and neuron, they're the same thing but different scale. Neuron is very small. Nerve cell is nerve cells is a big wire. So name the nerve cell that make up the nerve tissue. So basically that's what they are looking for. State 2 ways in which the brain is protected. This one we have been talking about this. All right. Brain is protected by 3 ways. Number 1, by the cranium. I've been saying that don't say skull because there is a difference between the skull and the cranium. So you must say cranium because the cranium is the brain box. Skull, it includes even the mouth, teeth, the jaws. Basically, I'm talking about the jaws, all that is a skull. But what they are looking for here, they're looking only the brain box, the part which protects the brain, because the teeth don't protect the brain. No. Yes. All right. Number 2 is, we say that meninges. Any there? Meninges. Yes. Number 2 is the cranium. Cranium. Yes. Meninges. These are the 3 membranes which are found in the brain. Around the brain, which you call the piamara, dioramata, arachnoids. We don't talk about them. Just know that meninges. Then you go to cranium, and then cerebrospinal fluid. Those are 3 ways in which the brain is being protected. Yes, it's simple. Even distinction material says it here. Cranium, meninges, cerebrospinal fluid. Simple. Someone is saying that you missed 1.26. 1.26. Serious? Nadia. Okay, let me check it. Let me check 1.26. Nadia is saying that I missed 1.26. Let me check it. 1.26. 1.2. Where is it? 1.26. A group of cells. Oh, thank you very much, Nadia. You're saying a group of cells in the pancreas, yes, that secrete insulin and glucagon. How do we call those cells? We call them isolates. This one disturbs the students with the spelling. Isolates of lung. When a penny. Okay. Isolates of iron. Okay, let me check now. This one is acting. doesn't know that I'm the best actor. Okay, isolates of lung, yes, Langhans. Yes, we call them isolates of Langhans. Yes, those are the special cells. Yes, we these isolates of Langhans, they have what called alpha cells and beta cells. Yes, but they want the general name because this is terminology. You can't say alpha and beta cells. No, isolates of Langhans. Isolates of what? This is the ear. Yes, we call it the ear. All right, let's go to the ear and then we find out what is it. Label it before you try to answer. Number 1, A is what? Tympanic membrane or tympanum. B, what is it? These are all the 3 oscars, all the 3 bones which you call the oscars. What is C? C, check the way the shape is oval window. Then what is E? E is this one. E is this one. It's the same as this one. This one is the same as this one. They just separate them. Yes, the inner ear, the middle ear, and then the outer ear is that side. So E is a round window. What is D? D is eustachian tube. What is F? F is semicircular canals. Yes. All right. Let's start now. Identify F. We have said it. Semicircular canal. Then give the collective term for bones B. We say that we call them oscars. Yes, oscars. Yes. They're saying that. So we say that these are oscars. Yes. They're saying that give the name, give the letter and the name of the structure that that what equalizes pressure between outer and the middle ear. That is the tympanum. Tympanum. So they're saying letter and the name. Letter and the name. Let's go back and find out the tympanum. Tympanum is A. So you can say that the answer here is A. Oh, they're saying equalizes pressure. Oh, no. They're saying equalizes pressure. The answer is D, not tympanum. The answer is D. D, which is eustachian tube. Yes, the answer is D. Yeah, D, it equalizes pressure on the either side of the tympanic membrane. They're saying equalizes pressure between the outer and the middle ear. Outer, this side of outer, meaning that this side of the tympanic membrane, and then middle, this side of the tympanic membrane. Hence, we shall say that it equalizes pressure on the either side of the tympanum. So, meaning that is D, which is eustachian tube. Number B is create pressure wave. Creates pressure wave. That is in the inner ear. It creates pressure wave. What creates pressure wave is the oval window, because when this they start to change, it causes this oval window to move in and out. So when it moves in and out, it causes this fluid which is found here and then it moves. Let me show you using the distinction material. Maybe you'll understand it better. Yes, here this is what I'm saying. Yeah, here is the ear. Yes, this is what they are saying. This part. You see this part? Yes, this part. It creates pressure wave here. This part. So it creates that pressure wave. You see now the pressure wave is going in. Ne? Yes. So this is the eustachian tube. They are saying it equalizes pressure on this side and this side. And then which other question? Other question they are saying that yeah, those are the two questions. Create pressure wave. So the answer is C and the oval window. C, which is equal to oval window. Yes, the paper was easy. Anyway, we shall see what as we are going down. Yes, we shall see as we are going down. Name the receptor that stimulated by the change in the position of the head. If you use the distinction material, what you call the SAIDI's model of balance. There we go. Change in position and head. Maculae. Change in speed and direction. Christi. What are they saying? Name the receptor that is stimulated by the change in position of the head. Maculae. Speed, direction and speed of the movement of the head. Cristae. Speed and direction, cristae. And then position of the head, maculae. Done. So when you are going to the paper, they will try to trick you. What? There is no way this book distinction material will never answer your question, will never make your life simple. It will always make your life simple, because I know that humans love simplicity life. Anyway, let's go to the next question, which is section B. It means that now you have 50 marks. 50 marks. Out of 50, you have 50 out of 50. Even if you don't do any other section, it means that you have already passed from the 30%. 45, you have passed. Out of 150, you have passed the 30%. But we are not looking for 30 marks. We are looking for a distinction. Hence, distinction material book was designed. All right, let's go to the next question. Hey, this is the one. What is this? Obvious. This one knows. If you're a boy and you can't answer these questions, then I don't know why. I know that boys like female reproductive system, and then you find out that girls, they like. If you ask the girls which system should they bring, they will say they must bring the male reproductive system, because I know it. How can you fail to know your system and you know someone else's system? And when you ask the boys, which one should they bring? They must bring the female. I hope now all the boys, you have managed to pass all these structures. Okay, A, what is A? Fallopian tube. B, endometrium. C, vagina. That one is obvious. What is the second name for vagina? Birth canal. All right, what is D? D is uterus. Yes, don't forget that the uterus is, I always say that uterus is divided into 3. The first one, uterine wall, endometrium, the inner lining of the uterus, which is the endometrium, and then the uterine cavity, the space of the uterus. Yes, so basically all of it is uterus, but it's just divided into different components. All right, let's go to the question and see what are they telling us. First question, they're saying B. Identify B. Obvious, we say that. Endometrium. Simple. Endometrium. Yes, endometrium. The second question, they're saying that name the process that takes place in part A. Where is part A? They're saying name the process. So what happens in part A is fertilization. The sperm finds the ovum there. Yes. So it means that when we go to that, we're gonna say that the process here is fertilization. Fertilization, yes. Describe the process named above. 1 mark. Describe the process of fertilization. 1 mark. It means that you have to make it as simple as possible. Fertilization is when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. Simple. You can't exceed that because it's just 1 mark. Yeah, but unless if it was 4 marks, then you can talk about the head of the sperm having the acrosome, which contain the nucleus, which contain the enzymes which digest the membrane of the ovum, so that the nucleus of the sperm enters the nucleus of the ovum to form a diploid zygote. But for 1 mark, you just say that is when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. It seems like they wanted you to define what is fertilization. Simple. Think that is clear. Yes. Describe the development of the zygote until implantation. This one has been my song to my students. Yes. Most especially the students who are always on ThunderEDUC. Yeah, ThunderEDUC channel. I've been telling you, and those students who have been attending my classes online. Yes. Yes. This has been my song. You can't go to the paper without knowing the development of the zygote until implantation. Let me show you from the distinction material. Yeah, what we talked about, the development of the zygote. Is there? development of the zygote until implantation. Even I showed you how do we mark this, ne? Development of the zygote until implantation. Exactly, that's what they are saying. Describe the development of the zygote until implantation occurs. There we go. Development of the zygote until implantation. They even didn't change any statement. There we go. The zygote, you saying a zygote, you do not earn a mark. Why? Because, yes, they mentioned the zygote. If they say that, describe what happens when the sperm fuses with the ovum to form until implantation, then mentioning a zygote will give you a mark. It undergoes, so we shall say that zygote, you're going to start with the zygote. The zygote undergoes mitosis. Yes, that's the first mark, mitosis. Mitosis to form a ball. You saying a ball is a mark cell called morula. Morula is another mark which divide mitotically or repeatedly to form a halo. The word halo is a mark. Ball of cell called blastula is another mark. And then this blastula forms the outer layer which you call the chorion. And the chorion forms what you call the finger-like projection called chorionic villi, which is used for implantation. I explained this one even before. Simple. Life science is easy. If you want a distinction, go do life science. If you want a distinction, don't just do life science. Use distinction material book. Where to get it? Download it in the link below. Yes. Thank you very much. Let's go to the next question. Describe the development. That one, 4 marks. Mahala. Yes. Explaining 2 ways in which structure D is structurally suited for gestation. Now ask yourself, what is structure D? Oh, the uterus. The uterus. How is this uterus structure suited for its function of gestation? First of all, what is gestation? The period of pregnancy. Yes, is what you call gestation. So, how is... Don't forget that you start with the sperm. Ne? Sperm. Yes, and the ovum. Yes, and the ovum. So, when the sperm and the ovum, they fertilize, you form a zygote. So, a zygote. Now, that is very small in such a way that you can't even see it. Yes, you can't even see it. So, but how big is the baby? Very big. Ne? It means that it must have that ability to accommodate this invisible, from invisible to the most visible individual. So, now in this case, it means that the first thing you can talk about here is it stretches to accommodate, is able to stretch, ne? Or the muscles are able to stretch to accommodate the growing of the baby. That's the first point. Stretching. Why does it stretch? To accommodate the growing baby. You see? Number 2, these muscles, they don't only stop there from stretching. Yes? Yes? They also contract during the period of giving birth. So it brings about labor during delivery, so that it facilitates this process of giving birth. So, labor, they can contract and relax, creating labor, yes, helping delivery process to happen very fast. Number 2, stretching to allow this space. You know this space? We say that this space is what? Let me show you. Yeah, how do you call this space? We said it's the uterine cavity, the space. So when it's stretching, the space increases. You want to know that the space is increasing, you will only see the tummy becoming bigger and bigger. Yes. So, it means that the thing is what? Is stretching. Anyway, what happens at night, we see it during the day. Yeah. So, let's continue to the next question. Describe how secretion of prostate gland, secretion of the prostate gland provide protection for the sperm from condition in part C. What is part C? Vagina. Now, ask yourself, how about the vagina? Is acidic. So, is acidic. Remember, if a vagina is acidic, yes, acidic. Yeah, what does it mean? It means that Sperms don't like acidic condition. So what should we do? The male reproductive structure, that is prostate gland, is supposed to produce an alkaline fluid. Yes, this alkaline fluid will neutralize. You see, it will neutralize the acidic nature of the vagina, and then it will be protecting the sperms from being denatured. Don't forget that sperms are protein in nature, ne? Yeah, so it tries to neutralize it so that it makes these sperms known to be killed by this acidic condition of the vagina. It is good for the vagina to be acidic, but it's not too good if it is too acidic. Yes, don't forget that anything which is too much is always bad. Anyway, let's go to the next question to do that question. These questions which are in a passage form, these are questions which I don't usually expect students to fail, because we don't usually bring them back the way they are. We don't. But you have to know how do you obtain information from the extract. Okay. You have to read the passage, ne? Very well, ne? Yes. So for the purpose of time, pause the video. If you are not on live, read it, or I hope you have a paper. Then after that, we continue. State one function of FSH, but they talked about FSH here, ne? Yes. State one function. What is FSH? You won't know what is the function of FSH if you don't know what is FSH. Follicle stimulating hormone. Sorry. We call it follicle stimulating hormone. A hormone which stimulates the development of the Graafian follicle. Simple. The name is stating that this hormone stimulates the development of the Graafian follicle. What is the state one function of that hormone? Its name is stating the function. Is the hormone which stimulates the development of the Graafian follicle. Simple. Done. Don't even waste too much time there. Name one other hormone that is in female, and this hormone is secreted by the pituitary gland during the menstrual cycle. Luteinizing hormone. Yeah, if you use the distinction material book, we have been saying this over and over and over again. Explain how. Under secretion of the hormone you have mentioned above may lead to infertility. So now you have to ask yourself, what is the function of that hormone? If those functions do not take place, will fertilization take place? If no, then you have to link to that question. So the first answer is luteinizing hormone is responsible for triggering ovulation. If ovulation is not triggered, do you have the ovum? No. If you don't have ovum, can you become fertile? Can you become pregnant? No. So it means that explaining how the under secretion of this hormone, meaning that you have the hormone, but it's low. It means that there will be low chances of the ovum being released, or there will be low chances of the ovum being triggered to be released, meaning that we don't have, then it will be low chances of fertilization. You understand? Yes. And if there is no fertilization, of low chance of fertilization, it means that you don't have the baby. What about when this luteinizing hormone, the second function is stimulation of the conversion of empty Graafian follicle to corpus luteum, and then now this corpus luteum, not corpus callosum, corpus luteum is not converted to. What is the function of corpus luteum? Is to produce progesterone. You don't have progesterone, no pregnancy, because progesterone maintains the pregnancy. Are you fertile? No. because you can't give birth. Okay, you can become pregnant, but you can't give birth. Are you fertile? I don't think so. So it means that there will be no progesterone, meaning that pregnancy cannot be maintained. Okay, progesterone will be there, but the chances, because we said under secretion, progesterone will be there, but it will be it could be less. But the major important thing here is the ovum. The chances of the ovum to be released are low, meaning that there will be low chances of the ovum being produced, and then low levels of that person to become fertile, to become pregnant, meaning that the chances of fertilization take place are also low. It's like someone having low sperm count. If you're not above 18, switch off your audio. So above 18, it means that even if you are eating in the morning, evening, supper, night, you can't have a baby, as you have to be every time you are struggling for the baby. When I say eating, I'm only talking about people who are old enough to understand the concept of what I'm saying. Yes, it means that if you are fertile, it means that you once someone is pregnant, once. If you are not, you have low sperm count. Hundreds of times. Explain why decrease in the maturation of ovarian follicle may lead to reduced implantation rate. Here, ovarian does not mature. How is that lead to reduced implantation? So you have to ask yourself, what does this Graafian follicle bring about? It brings about this hormone called estrogen. And estrogen, if estrogen is not there, what happens? It means that, what's the function of estrogen? It prepares the uterus for implantation. Check. If it's not prepared, reduced implantation. Prepares the uterus for implantation by making the endometrium wall, by making the uterus thick, vesicular, and granular. I stated it here very well in the distinction material. Yes, you can go to reproduction, or you can come even here. I stated it here very well. Estrogen prepares the uterus. Look, prepares the uterus for implantation. Check. By how? By making it thick, vesicular, and granular. It means that if we don't have this, it means that of this estrogen, first of all, first point, estrogen will be produced in less amount. That's point number one. You get your tick. Cool. Number two, you talk about the function of the estrogen. Estrogen will not prepare the uterus fully for implantation. Another tick. How? It will be less thick, less vesicular, less granular. I don't know how many ticks are there. Yes, less thick, less vesicular, less granular. And then, if the endometrium or the uterus is not ready, there will be less chances of implantation to take place. Yeah. So, I call this book My Baby. Why? It solves all the problems of my babies. When I say my babies, I mean my kids from the other fathers and other mothers. I mean my students. All right. Let's go to the next question. Describe the process of spermatogenesis. Distinction material. Don't forget that the process of spermatogenesis is not the same way the other books, I'm not going to say those books, are stating it out. We modified it based on the new examination guideline. That's why this book is updated every year. So if you have a version for the last year, you have to use the version for this year, so that those small things which we keep on adding, you can do what? You can be corrected. Spermatogenesis is the formation of the sperms. Process. Check what they are looking for. How many marks? 4 marks. Check. Under the influence of testosterone, 1 mark. The diploid cell, another mark, in the seminiferous tube will undergo mitosis to form 4 haploid sperm cells. Simple. 4 marks are there waiting for you. I told you that different color means a marking point. Under the influence of testosterone, yes, the diploid cell in the where are they found? In the seminiferous tube will undergo meiosis to form 4 haploid cells. Even if you say to form haploid sperm cells, correct. Simple. Yes. Yes, we're no longer using this method. Yes. I was showing you here so that you understand exactly the science behind, but this is what we want. Clear? Let's go to the next question. Frogs. I know some of you like frogs, some of you don't like them. Yes. Frogs survive in water and on land. Most frogs, however, need water for reproduction during breeding season. Males and female frogs release millions of sperms into the water. They're saying 2.4.1. Name the type of fertilization and describe described above. Fertilization, look, where is it taking place? Millions of sperms are released in the gametes into water, meaning that it's not inside the female. So the answer is going to become external fertilization. Yes, external fertilization. Explain why millions of sperms are released. Yes, why do we have to release millions and millions of sperms? So to increase the chance of these sperms to meet the ovum so that fertilization can take place. Yes. So why do we release? To increase the chance of these sperms to meet the ovum so that successful fertilization or fertilization can take place. That's what they are looking for. State why reproduction in frogs is an example of oviparous reproduction. What is oviparous? Yes, oviparous or ovipary means that the development of the embryo is outside the female's body. So it is classified as ovipary. Why? Because the development of the embryo is out. It's not about fertilization being outside. But definitely, if fertilization is outside, do you expect the egg to go back inside the female's body? No. So the development of the embryo occurs outside the female's body. Next question. So those marks must be obtained. The skin. I used to draw the skin, and then some people were saying that the skin is so simple. Actually, the skin is so simple. Yes, very simple. They're saying that, okay, what is A? A is the sweat duct, sweat gland, duct, sweat. Add the word sweat, add the word sweat, pore, sweat, outside, sweat, sweat pore, sweat duct, sweat gland. Then these are capillaries. All right. Yeah, those are the things which you need to know about the structure of the skin. Give 2 reasons why A is classified as exocrine. Why is A? First of all, exocrine, they have ducts. We call them duct glands. That's what I said here in the distinction material, that we call them exocrine because they have what we call the duct. They are duct glands. Yes. Check. Exocrine glands, we call them endocrine glands, we call them ductless glands. And then these exocrine glands, they what? We call them duct glands. Yes. And then number 2, sweat gland secretes the sweat where it is needed, outside the what? The skin. Therefore, the secretion is secreted directly to where it is needed. Simple as eating ice cream, no bones. Yes. Describe the role of the skin in the thermoregulation. How is the skin? Skin receptors? How is the skin receptors? Yes, important in thermoregulation. Basically, the skin receptors, they detect the change in temperature and converts the stimulus into the impulse. Simple. Detect the change in temperature, and then they convert that stimulus into a nerve impulse, which is sent along the neuron. So now, detection is a marking point, and also conversion of the stimulus into a nerve impulse is another question, another mark. Explain why structure B dilates on hot day. Why do we have a big dilation here on hot day? Yeah, why do you see your blood vessels on hot day? Yeah, the same thing I was talking about on the skin. Guys, there is no way in this life science you're gonna fail this bone. There is no way. Yes, what you need to do is to do what I'm telling you. Download the distinction material book, use it, make sure that you get a distinction. I'll be happy where I will be, whether I know you or I don't know you, as long as you're happy. All right, let's continue. They're saying that yes, here check how they are. They are big, ne? Yes, they are big. You see, much heat is lost, so it dilates so that it can allow more blood to flow near the skin. You see, more blood is flowing near the skin. More blood is flowing near the skin, and as it flows near the skin, heat is lost. That's what they are trying to say. So allows more blood to flow near the skin so that more heat is lost. Yes, that's it. That's why it dilates during day, hot days. Sometimes we can also say that it dilates so that it can supply more blood to the sweat gland, so that the sweat gland can become more active. What are the work in some factories are constantly exposed to loud noise. Okay, this is just a story for a long period of time. This can destroy the hair. Okay, it can destroy the hair cells in the organ of Corti. They can destroy. Okay, we know that now the loud noise. People who are staying in towns where beep 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 beep. Hey, this one is making noise. This one is putting on music. Take note of that. Damage and the auditory nerve, resulting in hearing loss. Okay, survey was conducted in the developing countries. 2014 to 2018, to establish the number of factory workers who suffer from this hearing loss. Results are shown in the table below. Question number 1. Name the structure where the organ of Corti is located. Cochlea. We call it cochlea. Yes. Calculate the percentage increase. Yes, percentage increase. We like to ask you this. I don't know why. Maybe students, they don't get it right. Yes. Here percentage increase is equal to final minus initial over initial times 100%. Final, what is final? 2018. So my final is gonna be this 130,000 minus initial, which is 85,000. Yes, divide by the initial 85,000 times 100%. percent. So you calculate using your calculators. What is the answer? Around maybe 50, maybe 52.9%. Yeah, so that becomes the percentage increase. Sometimes they can say percentage decrease, same thing, but that might be, yes, it must be maybe in negatives. Anyway, let's go. They're saying that suggest one reason why increase in the number of factory workers with hearing loss caused by exposure to loud noise in this can suggest one reason for the increase in the number of factory workers with hearing loss caused by exposure to loud noise in this country. They said it here. They said it here that this can destroy the hair cells in the organ of Corti and damage the nerve to the nerves. Yes, they said it here. That's why I like the passage. You get the answers from there. Draw a bar graph. Bar graph, you want me to draw? I don't think so. But what is important here is you have to be with a caption with the two variables. Yes, meaning that you have the variable of the year and the number of workers with that problem. And then we use the ID method. ID method. You remember the ID method? Remember the ID method? Some of you don't know the ID method. Let me show you the ID method. All right, the ID method is, you say ID X Y, independent variable on x-axis, dependent variable on y-axis. This is the independent variable, and then this is the dependent variable. Simple. And then now it means that number of workers is going to be on the where y-axis here, and then number of years is going to be where here. Simple. And then if you look at this, because there is increase, you will find out that it will have that shape. Don't forget, how do we mark? This must be years, but years. Yes, if it has units, make sure that you put units. If there's no units, the same thing. And then your numbers, you must put the units. If it's there, make sure that the distance between here and here and here and they are the same. Make sure that these bars, they have the same breadth, meaning that how big they are must be the same, or otherwise you're gonna lose marks. Yes, so we mark the caption, we mark the correct type of the graph, because some people, they will draw a histogram instead of a bar graph. We mark the plotting, we mark the axis. Yes, we mark the axis, x and y, and then also mark the scale. Scale, make sure that if you start 0, 2, 4, for in this case, it's gonna be what? It can't start from 0. Yeah, why? Because it's not that big. Maybe you're gonna start from 5,000, 50,000, 50,000, 100,000, something like that. Or you will see the best scale you can use. Yes. So that is the question. Hope I'm clear there. Let's go to the next question. Which question is this? Question 3, meaning that now we have 100 marks. Mahala. 100 marks. Yes. Meaning that 100 out of 150, how much is that? Yes. So now you know how much you have. Yeah, tell me, is the paper easy, difficult? Let's go to the next question. They are saying that the graph below shows the concentration of abscisic acid in the cherry tree during the different seasons of the year. They are saying that this tree species loses all the leaves in the autumn and goes into state of dormancy, meaning that this tree, in science we call them deciduous plants. Plants which lose their leaves during the period of what? During the period of winter. Yes, we call them deciduous plants. Yes, question 1. Question 1. They're saying that I need to be a little bit faster so that I finish. The first question, they're saying that during which month was abscisic acid concentration lowest? It's obvious, guys. During which month? Check. This is concentration. November. Simple. November. Explain the trend of the graph from March to May. They're saying explain, not describe. Why? From March, they said March, from March to May. When they say explain, you have to give a reason. Yes. From March to May, there is an increase in the concentration of abscisic acid. Why? Because, don't forget that they say that abscisic concentration, different season of this tree lose all the leaves during autumn, meaning that the abscisic acid is the one which is important for shading of the leaves. So it means that why does it increase? So that it triggers that shading of the leaves, so that the plant can undergo dormancy. So you mentioning increases, yes, a mark, and also explaining why does it increase, so that it can bring about the shading of the leaves during this period, so that it enters when it is already dormant. Does not lose a lot of water during winter, because it doesn't have that water in the system. All right, state one reason why dormancy in cherry trees. Suggest one reason why dormancy in cherry trees during winter month. Why do they shed? Why do they undergo dormancy? So that it minimizes the amount of water loss to the atmosphere. Yes, don't forget that water loss is most especially through leaves. So when you shed the leaves, it means that you are minimizing the amount of water being lost to the atmosphere during this period of dryness. Yes, the next question. Geotropism. Geotropism. Yeah, how about geotropism? If you look at geotropism, what are they looking at here? Geotropism is a plant response to the gravity. Sometimes it's called gravitropism. Movement of the plant response to gravity. Oh, they have talked about this. Tropism is controlled by auxins. Describe the role of auxins in the roots. Basically, auxins are found at the tip of the root. And auxins, they bring about growth. So the first role is it brings about growth. But how does it bring about growth? High concentration of auxin will inhibit that part which has high concentration of auxin will inhibit growth. So basically, that's what you talk about, meaning that the part which has low concentration of auxin will grow faster, and then to bring about the bending action of the root. Then they're saying that 3.22, when a plant is placed horizontally, okay, you place it horizontally with light coming from all the direction, auxins will accumulate on the lower side of both stem and root. Explain the difference in a response on the root. And okay, here they are asking you that if you have light, you place the two things horizontally. Let me show you here in a different what here. They are saying that okay, you have a plant, you place here and you also place here. If this is a shoot, and then this is a root, yes. But if you have light from top, yes, from top, what's gonna happen? You're gonna have high concentration of auxin here. High concentration of auxin here. Don't forget that the role of auxin in roots, it is brings about growth. Even here brings about growth. But in roots, high concentration brings about inhibit. And then high concentration in root brings about promote. It means that this side is gonna grow faster, bringing about bending upwards, while this one is gonna grow slower and this one is gonna grow faster, bringing about the bending activity. Yes. So basically, in roots, the leaf, the shoot will bend towards the source of light, whereby the high concentration of auxins will stimulate that growth. And then in roots, the root is going to bend away from the light, meaning that high concentration of auxins in root will bring about inhibiting. So this is what I'm saying of the inotropism. If you look at our distinction material book, yes, that's what they are saying. High concentration of auxin bring about that bending towards, you see. And then in roots, high concentration, check the high concentration is here. It brings, you see, high concentration is here. It brings about the bending. The one which has no high concentration bends, grows faster than the one which has. That's why you see these ones that are too small, while these ones are longer. Don't forget that growth is elongation of the cell. Yes. So basically the upper part will go faster than why? Because the auxins will be high at a lower side, meaning that the upper side will grow faster than the lower side. So that's what they are talking about there. The second question is so here what they are looking for is to show us that in roots, high concentration promotes, resulting in the bending of the shoot to a source of light. And in roots, high concentration result in inhibition or inhibiting or limiting growth, resulting in the root bending to the ground or away from the light. Yes, let's go to the next question. The next question is talking about, what is talking about? Yes, independent variable and a dependent variable. Here, I'm not going to waste too much time because we always ask you questions concerning about this. We don't bring the same question, but I'm seeing that my battery is dying. All right, let me just say, name the gland that secretes aldosterone. Let's just go straight to the question without, you have to read here. But what is most important here is this. Scientists investigated the influence of decrease in aldosterone level on the blood. Yes, scientists investigated, yes, aldosterone level in the blood. So this is the most important thing. Yes. So if that is the most important thing, so it means that it can help us to know which one is independent, which one is dependent. Identify independent variable. Aldosterone is secreted by adrenal gland. Adrenal gland. Yes. And then independent variable, they are saying that investigated the influence of increase in aldosterone level on the blood pressure. Blood pressure versus influence of increased aldosterone level. This versus this. So it means that blood pressure is being influenced by this. So blood pressure becomes dependent, and then the influence of aldosterone level is dependent. Then the next question is saying, give two reasons why results may be considered reliable. The reliability, don't forget that you repeat the experiment. Did they repeat the experiment? Yes. Let's check. Yes, they said that they repeated the experiment. They measured every, this is average. Ne? Is it average? No. They measured every 4 hours. Every 4 hours for 12 hours. Meaning that they kept on repeating. And then they said that they calculated the average pressure. That is average. So meaning that they calculated for average, and also they repeated over a period of 4 hours for 12 4 hours for 4 hours for every 4 hours, for every hour for 12 hours. They repeated it 12 hours, 12 times basically. Yes. They're saying that explain two reasons why there was important for participant for the same diet. Yeah, so that you have the same maybe salt concentration, and then also to keep the same blood pressure, because if you have different types of food, it will bring, because they said that it brings about blood pressure. Yes, somewhere there. So I'm just rushing so that I go to the last question, because that is an open question. This is not that we're gonna bring the same question, ne? Yes, so it's just how do you extract it from the question. They're saying explain why participant blood pressure was measured before the start of the investigation. Whenever you do that, it means that you are looking for the reference point, basically to act as a control, or to have a reference point where you know that, okay, without it, or without it, what will happen, or before you applied the experimental measures, what was the conditions before, so that now you can bring about comparison. So basically is to compare the results. Or reference point. Explain why the level of salt in the urine of the participant expected to decrease after being injected with the aldosterone. Don't forget, the function of aldosterone is to stimulate the nephron so that there is more reabsorption of this salt back to blood. It means that you have less salts in the urine, and more will be reabsorbed back to the next question. Yes, is the last question, I think. That is the eye. I know you like the eye, ne? You like the eye the way I like it. Yes, I think you like the eye the way I like it. So they're saying that what is A? A is vitreous humor. B is yellow spot. C is blind spot. D is lens. E is cornea. F is iris. So you have to know them. Identify F. We have said it. F. It is iris. And then they're saying state two functions of A. Where is A? Function of A. It brings about the shape of the eyeball. Yes, it brings about, ne? Yes, it maintains the shape of the eyeball. I stated it here in the distinction material book very well. If you have the eye, then it means that you have this book, as I told you that there is no way you're gonna fail. Yes, check it here. I said it here. This is aqueous humor. Change, yes, changes the shape of the near and far distance. No, that's the lens. That's not is here. It gives the shape of the eye. It gives the shape of to the eye, meaning that it brings about that shape of the eyeball. Then number two, state the function of A. Okay, we have seen it. Describe the structural difference between B and C. Structural difference between B and C. B is yellow spot. That's where the clearest image is formed. So it has more rods and cones. Highest concentration of rods and cones are found in B. They are saying structure. And then C has no photoreceptors, has no rods and cones at all. Meaning that if the image forms there, you'll be blind. You won't be able to see that object. That's why you're saying that it is the blind spot. The next question is saying that describe this. Okay, name the visual defect occurs when the curvature of the eye is uneven. When the curvature of the eye is uneven, we call it the curvature of the eye is uneven. So we call it astigmatism. So we said it here. Astigmatism is when the cornea or the lens is not evenly rounded in all directions. It means that it's not evenly distributed. So it is called the astigmatism. Then they are saying that explain why sight on person, explain how the sight of the person will be affected if cataracts develops. Remember, we said that cataracts here is when the eye is cloudy, the lens is cloudy. So it means that light will not be able to go through. The lens becomes opaque, you see. So it means that you will be having this blurry vision. Yes, that's what they are asking you. What will happen? You will have blurry vision because light rays will not go through the lens. Why? Because the lens has become opaque. Check. Use distinction material. There is no way you're gonna fail. Yes. The next question, they are saying that describe the process of accommodation that takes place when the object is less than 6 mirrors. bafethu. Still bafethu. This paper bafethu. Ne? It is less than 6 mirrors. This is more than 6 mirrors. So memorize one and then do the rest. Just have to memorize one. We are saying that ciliary muscles contract, suspensor ligament slacken, tension on the lens decrease, the lens becomes more convex, light rays is reflected to the retina, or more light rays are bent more, and then a clear image is focused onto the retina. I'm rushing because of the electricity is gone. However, my system is dying in the next 3 minutes, yet the electricity is coming back within 7 minutes. So it means that I'll be having that time when I don't have electricity. So you guys, I'm going to say that thank you very much for being here with me. Yes, I was discussing the final paper for 2023 Life Science. You saw that this paper was easy. Yes, this paper was really easy. Most especially those people who used my distinction material. Yes, those people who used my distinction material, I know that you got this paper. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Another disturbing question to students in paper 1 of life science. The development of the zygote until implantation. Students really struggle with this question. But before this video ends, you'll be able to write what is needed in the paper, and then you score all the marks. So development of the zygote until implantation. Let's get started. Let's first describe all the stages which are involved in this process. So you start with the sperm and the oocyte. Oocyte is the ovum. So you start with the sperm or spermatozoa, fuses the nucleus of the sperm fuses the ovum, and then when it fuses, it forms what's called a zygote. So it means that here you have the nucleus of the ovum, and then you have the nucleus of the sperm. It's not the whole sperm entering, no. It's just the nucleus of the sperm. That's why we are saying that haploid nucleus of the sperm, not the sperm entering, no. Haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. So a zygote is one cell, but is diploid. It has 2 sets of chromosomes from the father and from the mother. While the sperm is haploid, has 1 set of chromosome, and then the ovum has 1 set of chromosomes. So this is haploid, this is haploid, half plus half, you form what's called a diploid. So the zygote must undergo a process which we call mitosis to form different cells. It undergoes mitosis, like 1 cell divides to form 2, 2 divides from 4, 4 divides from 8, like that. So it undergoes mitosis repeatedly. So it forms what we call a ball of cells. So when it undergoes mitosis repeatedly, it forms what we call a ball of cells. We call it morula. This is how a morula looks like. It's like a ball. That's why you say that it forms a ball of cells called morula. So we are saying that this ball of cells undergoes mitosis continuously, forms a space inside. This space, so it becomes hollow. It means that it has a space inside. So it forms a hollow ball of cell called blastocyst or blastula. Yes. So it was a ball, but now the ball forms a hollow, forms a space inside. You can see it here. See, the space inside is big. So this space, it makes it to be a hollow. So it forms a hollow ball of cells called blastocyte. So we are saying the blastocyte now, it forms the outer layer. This outer layer, yes, we call it now the chorion. This outer layer starts to form the finger-like projections. These are the finger-like projection, which you call the chorionic villi. These chorionic villi are the ones which are very important for implantation. So basically, that's how you can explain it. So we are saying that how do you write this in an exam? The sperm paralyzes the ovum to form a zygote. So we can write that
After fertilization, the zygote undergoes mitosis. So this zygote, it undergoes mitosis. When it undergoes mitosis, it has to undergo mitosis. One cell divide, form into two, forms four, form 8. So it forms a ball of cells. So we are saying that it forms a ball of cell. This is a ball of cell called morula. So the zygote, yes, undergoes, we give you a tick, undergoes mitosis, we give you a tick, to form a ball of cell. This ball, you have to write it that a ball of cells. To form a ball of cell called morula. So we give you a tick, a ball of cells called morula. It also give you a tick. So that's how you try to, each statement which is in different color, it shows that that is a tick in the exam. So it depends on how the question is set. But in most cases, if it is an open question, then it means that each color is a tick. The morula still undergoes mitosis. When it undergoes mitosis, then we are saying that it divides by mitosis repeatedly to form a hollow ball of cell called blastula. So if you look at it here, yes, it undergoes mitosis repeatedly. So to form a hollow ball, it has a space, as we explained it before, hollow ball of cell called blastula or blastocyte. So we are saying that it forms the outer layer called chorion. So if you look at it, yes, this ball of cells, yes, the outer layer, this outer layer, yes, this outer layer, it forms this outer layer which you call the chorion. So this chorion, it modifies, it forms a finger-like projection. Finger-like projection, we call it chorionic villi. Yes, it is a chorion, but now the finger-like projections, which we call chorionic villi, are used for implantation in the uterus or the endometrium. So basically, this is how it looks like. This finger-like projection forms what called a finger-like projection, which you call the chorionic villi. So this is the outer layer is the chorion. So this outer layer starts to protrude out. These protrusions or the finger-like projections, call them chorionic villi. These chorionic villi are very important for implantation. So basically, that's how you need to explain it. Let me just repeat it once and for all. After fertilization, yes, when they ask you to explain this, don't explain the process of fertilization. We don't need to know the process of fertilization. They want you to explain what happens to the zygote immediately, or what happens to the zygote until implantation. So you said that after fertilization, a zygote is formed, which undergoes mitosis. It undergoes mitosis to form a ball of cells, called morula. Morula undergoes mitosis, or it undergoes mitosis repeatedly to form a hollow ball of cells called blastula. So it forms a hollow ball of cells called blastula. It forms the outer layer called, this outer layer forms the outer layer, which called the chorion, which forms the finger-like projection. So it develops the finger-like projection, which we call the chorionic villi. What's the function of the chorionic villi? It's very important for implantation. So basically, that's what you need to know, nothing else. So you just have to know that this is a zygote. This is an ovum. It is haploid. This is the sperm is haploid. The first cell is a diploid, because haploid from diploid. This is also a diploid. Yes, this is also a diploid. Also, this is a diploid. So basically, these are undifferentiated cells. They can develop into other structures. If you look at it here, it's just trying to show you the 3D presentation. That this is the egg. Yes, the egg after being fertilized. What happened? The egg immediately after fertilization. So yes, this is the egg. It undergoes mitosis repeatedly to form a ball. Yes. From here to here, it is all that is mitosis happening, which you call morula. Morula undergoes mitosis to form a hollow ball. Inside there is a space, hollow ball of cells called blastula. Then blastula, yes, forms the outer layer, which you call the chorion, which forms the finger-like projection. Yes, finger-like projection for implantation. So basically, that's it. And then here are some of the questions. Identify the process taking place at 1 in the diagram above. So if you look at it, sperm fuses the ovum, so it is fertilization. State the type of cell division that takes place in 2. You see, we talked about it. So the type of cell division is mitosis. Then there's and give 2 functional of extraembryonic membrane produce functional extraembryonic membrane. So we say that it forms a chorion. Chorion, yes. Then also we have what we call the amnion. So we have not talked about amnion because we have not yet reached there. So it forms the chorion and then the amnion. Identify the stage that of development indicated by A. So A stage of development, this is just a zygote, and then B is a morula, and then C is a blastula. Name the part of the female reproductive system where events in the diagram above usually takes place. Fallopian tube. Look at it here. It occurs in the fallopian tube. So this is how the sperm fertilization takes place here. So this is the development. So implantation occurs here, exactly here. So this is the ball of cells, and this is the hollow ball of cells. Within a maximum of 1 week, this process has taken place. This is the time when a person has started to show the signs of pregnancy. Signs of pregnancy. Someone is weak, someone is vomiting. This is the period. Why? Because the body is experiencing new things. So we can also set it in this format, and then we ask you what is really happening here, describe the process, you see. So until implantation, from this point after fertilization, then it goes until it reaches here. So next time we will look at fertilization in detail, and then I will show you how we can also write it without having any problem. See you next time. Subscribe if you have not yet subscribed for the new videos.
On what we call degrees. Why? Because the circle is made up of 360 degrees. So you cannot plot it in percentages, mass, what must be in degrees. The first thing which you need to have is a protractor, because there is no way how you can draw a pie chart correctly and then when you don't have a protractor. The protractor has two scales. The first scale is the clockwise scale. The second scale is the anti-clockwise scale. There is this scale which starts from 0 here, and then it moves like this, comes here to 180. And then there is another scale, outside scale, comes from here, it moves, then it comes, it's 180 east here. So it depends on whether you are a left-handed person or right-handed person. The two options are there. Oh, you can be even left-handed and then use the other scale. So it depends on what you really need. So once you know how to use, so I usually call this one because it's moving anti-clockwise, the anti-clockwise scale. Outside one, usually call it the clockwise scale. So any scale you use, still you'll get it correct. The second thing you need to have is you need to have a compass. This compass, a pencil must be well fit. You must fit it properly into the compass. Then it has you must have what called a ruler. So the function of the ruler, the first function of the ruler is to measure at least 5 centimeters. The distance between the first tip, the tip of the compass, and the tip of the pencil must be 5 centimeters at least. Why? So that you can draw a bigger circle. Yeah, we shall see how we draw it. If this space between the tip of the compass and tip of the pencil is too small, for example, is here, the circle is going to be very small. Yeah, it's going to be very small. So this makes the work to be difficult to be marked. So the more you draw it bigger, the more clearer it becomes. So it also makes the marker happy when he's marking, because it's not going to strain the eyes. And so these are the steps we're going to be doing until we complete to draw the circle. We're going to see that practically, or what you call the hands-on. Then you are given a table which has all the information which you need to use for plotting. So sometimes this is not added, sometimes it's added. If it's not added, so you have to add them. For example, the total is going to be equal to 18 plus 9 plus 6 plus 3. So if it gives you 36, so these are not degrees. It depends on what they have given you. Yes, if it is in degrees, then you just plot, just do the pie chart without converting. But if it is in a mass, number of people, what percentages, you have to convert them. So to calculate for the 18, I'm gonna say 18 is blue. Blue is gonna be equal to 18 out of the total, which is 36 times 360, so that I convert it into degrees. So I bring my calculator. Then once I bring my calculator, it's gonna give me 18 divided by 36, answer times 360. I think it's 18. So this is going to be 180 degrees. And then I go to the next one is green. Green is equal to what is it? 9, eh? 9 over 36 times 360. You bring your calculator again, and then you say 9 divided by 36, answer times 360. It's 90. These are the degrees you're supposed to plot. The degrees. And then you also do for red and then yellow the same thing. And then end up having this. You also end up having that. Then once you have got these degrees, then automatically it means that you are ready to go for drawing a pie chart. So let's go for drawing the pie chart. You need to have a compass, a ruler. This is called a protractor. Yes, you need to have a pencil, and then you need to have a pen. So these are the things which we are going to use because we have already done our calculation. So these are the things we are going to use so that we can start drawing. So the first step is to put the this. If it doesn't go through, you open. Please don't go to the paper without having this. So you make sure that they're on the same level. Yeah, you see that they're on the same level. Yes, once you see that yes, they're on the same level, then you make it tight so that it doesn't move and doesn't shake. All right, so that's number one. Then step two is you're supposed to measure. Remember I told you that try to measure this, measure 5. So 0 to 5. The reason why I'm saying it is good to measure 5 centimeters so that when you draw, for example, you want to draw it here, you make this one tight, and then now you turn this like this. Make sure that the center doesn't change, because if it changes, yeah. So you see how big it is. It's big enough. If this is small, is smaller than 5, for example, is 2 centimeters, you're going to end up drawing something very small. See? Something very small. And then this is going to give you a problem when it comes to measuring the degrees. So we prefer you to use 5 centimeters so that at least the circle is big enough. And then it also makes the examiner happy. It makes the examiner happy when you are at least. So draw it, put it there where you want it to be. And then now start to draw. You can even just do like this, just change the paper like this. You change it correctly, yes, it will give you the correct circle. Or if you don't want to change the paper like that, you just draw, keep it, and then start. Make sure you use a sharp pencil, eh? Yeah, because we want some things which are sharp. Yes. So we want things which are sharp. So I'm done. That is the first step which you needed to know. That is the first step. So now the function of the compass is done. I can remove my pencil. Yes. If you see that this is not sharp enough, try to be with your sharpener just next to you. Try to make it sharp. Yes. And then now draw. You have to be neat. You have to be neat. So you draw a line going to the circumference. So it means that I've drawn what you call the radius. Now I'm going to bring my protractor. This is the protractor I said. I say that this protractor has two scales, has a scale from 0 to 180. So I will say that because it is moving anti-clockwise, therefore I'll call it the anti-clockwise scale. And then this one from 0 to 180, the outer one. So I'll call it the clockwise scale. Yes. So there is this part. Yes. This part is where it's supposed to be placed onto the, you're supposed to place it where the compass was. And then this line is supposed going through this line you have drawn. So I put it there. Yes. So I see that now it is in line. So now I come back here and then I check. The first degree is 180. So I come here and then I measure the first degree is 180. So I come here, it must be straight. So now if I use, because now I've started from this side, I'm going to use the inner scale because now I can't measure from that side. So because this is my first point, yes. So I measure 180 is gonna be there. So I put a mark there. Yes. So now after that, I bring this ruler and then I join it. I join it to the mark. So I join it to the mark. So basically that is 180. But because now I stopped here, so now I have to turn this paper. And then I look for the second one. The second one is 90 degrees. So now I do the same. Remember, this is our center. Yes. Now I put it here. Yes, I put there. So because I'm using anti-clockwise, you can use even clockwise. You can measure from that side, depending on your choice. So I come here, say 90 is here. Put it there. Then now I come and then I draw 90 is there. So it means that this is 90. Then I come to 60. Then I turn it. 60. Measure 60. Yes. 60 degrees. 60 degrees is here. Degrees is there. And then now I come here. I use my ruler. And then I draw 60 degrees. So, yep. Now, the remaining one must be 30 degrees. If it is not 30 degrees, it means that you did something wrong somewhere. So if I check here, you see that 30 degrees, it clicks in 30. You see, it clicks in 30. So basically, that's how you supposed to draw. Now, you can use a compass or you can just use hands, and then you tell us that these are those, how many degrees are those, then how many degrees. You can use a compass so that it looks, yeah, it looks nice. And then say that I need to know what is a key. Then I say, now I start to do whatever I'm doing. Maybe I call this one, let me just shade it like this. Yes, let me shade it like this. I come here and then I say what I've shaded like that. Now I've shaded it like this. And then now I come here and say that this is the blue. So this is the blue. And then I come here, maybe I call this one S, 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 S. Come here, the box and say S, S, S means what? S, 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 it is S, 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 it is the smallest. So this is 180. Yeah, 80 degrees. This one is 30 degrees. This one is was 90 degrees, and then this is 60 degrees. So basically, that is it. This is it. S is yellow, and then I can call it P, whatever you use or whatever color you use. And then this one PPP is the 60. We said is red. Then say this is red. Then say PPP, PPP is red. And then lastly, maybe I can call it XX, whatever you sign use. And then I come here and then I say XXXX, and XXX is what? XXX, XXXXXX is going to be is green. XX is going to be green. So basically, that is it. Now the next thing is you're supposed to write the caption or the title. Don't forget to write the title. The title is supposed to have the two variables. So now, how do we mark? Basically, yes, we will mark the type of the graph. It's a pie chart. Maybe give you a mark, and then we'll mark the caption. We'll give you a mark. We'll mark the plotting. We'll give you maybe 2 marks. Yes, we'll mark the key. We'll give you a mark there. We don't stop there. Then we come back here and we see the calculations you have done. Also come here on the calculations. Maybe we shall say that calculations also maybe we give you 2 marks for each. And then now maybe they say calculations, we give 2 marks. Then depending on the memorandum, how is the memorandum, what is the memorandum saying? So basically, that's how you're supposed to draw a pie chart. Let me know your problems so that we can solve those problems, because this was one of the questions for one of the students say that wanted to know basically how do we draw a pie chart. Thank you very much.
Positive feedback mechanism, but we don't talk about it here. When you go to the universities, you'll talk about it. But now here, when you talk about this negative feedback mechanism, it means that something which went wrong must be brought back to by subtracting what that made it wrong, then you bring it back to normal. That's why we call it negative. But if something get wrong and then it becomes more wrong, then it becomes positive feedback. But we don't talk about it. Yes. So here, this is just to bring about the normal functioning of the body so that everything works to the way it's supposed to work. So this enables the body to function efficiently. So when it works efficiently, there is a difference between efficient and effective. So don't use effectively, use the word efficiently, because when you talk about efficiently, yes, the body is supposed to work. When you say efficiently, sometimes the efficiency might not be about 100%. When you go to physical science, you know that better. But despite the changes which might happen in internal and external environment. When we talk about external environment, we're talking about the surrounding areas. What you have, maybe the temperatures of the surrounding, maybe you talk about the humidity, that's the surrounding. When you talk about internal environment, we are talking about maybe the blood glucose, the salts, the water. Did you know that a human being has the same temperature, however much this person goes to water or to the ice, the temperature remains the same? So that's what you're saying. Despite all the changes which might happen, you understand? Yes, the internal environment must be kept constant. So now, if it fails to be kept constant, then now the problem will come in. How does the problem come in? Maybe the system will fail to work properly. I think I'm clear there. Good day, sir. Hope you are well. Thank you very much for simplifying lessons, making it easier for us. Okay, thank you very much, guys. Yes. Make sure that you do what? You share to others also so that they can benefit. Guys, if you have a problem, this lesson is live. Ne? This lesson is what? Is live. So if you have any problem, let me know. I can try to answer those questions. I'll be giving you some questions. This lesson might go for 1.5 hours, if not 2 hours, so that we maximize all the things which you might make this situation to be difficult for you, so that you guys will get distinctions, because that's what we are looking forward for. All right, we are saying that changes in temperatures, glucose levels, carbon dioxide, and water levels, plus salts, these are the internal environments we are talking about, which are supposed to be kept constant. Yes, so those are the internal environment we are talking about. So this internal environment must be kept constant. Yes, so we are going to look at them in detail. So they are saying that regulation of glucose levels. Now here you need to understand some few things before you go to the regulation. Let me give you a trick so that you can understand this better if they bring it in terms of this diagram you see. Let's start with the glucose. Number one, glucose. Guys, you need to know two things from two things. The first thing you need to know is always in the middle here, you have what called the normal. Guys, all of the mechanisms, they look like this. You don't need to stress. So always it is like this, normal. You understand? When you say normal, okay, what is going to happen here? The last statement and the first statement must be in opposite way. If you see here there is increase, yes, the last statement must be decrease. If you see here there is decrease, yes, the last statement here must be increase. I think I'm clear there. Whatever negative feedback mechanism you're gonna talk about, except one, that is carbon dioxide. I'll explain that better regarding to why carbon dioxide is not made to be like that. It's because its mechanism is one way. It's not two ways like these ones. So now we are saying that the first one must be, if it is increase, the last one must be decrease. If the first one is decrease, the last one must be increase. So now you only have to fill in 2 points here. So make sure that you separate these 2 from these 2. So how do I know these 2 points? You need to know what are they talking about. This is also being spoken about here. The normal level of glucose in the blood. So if glucose level increase, the last statement must be glucose levels will decrease. If the glucose level decrease here, the last statement here must be the glucose levels increase. I think I'm clear there. Now, when it comes to glucose, then you have to ask yourself, when it comes to glucose, which gland is responsible for glucose? And what is the function of that gland? So you mean that if you're talking about glucose, glucose goes with glycogen. So glucose, yes, is being acted upon by which hormone? And which gland does it which gland produce that hormone? So it means that the hormone and its function will be here, and the gland and its function will be here. Meaning that the function of the gland is the hormone it produces, and then the function of that hormone will bring about the decrease in that level of whatever you are talking about here. For example, say that glucose level will increase, or definitely the last statement will say that the glucose levels will decrease. So that one is sorted. 2 marks gone. Oh, you have got them before even you go to the paper. Then you have here pancreas, because the glucose, what works on glucose and glycogen is pancreas. So the pancreas, that's the gland, will produce or will secrete insulin, meaning that you have talked about the gland and its function. Meaning that the gland, which is pancreas, what is its function? It secretes insulin. Done. Then you come here and you tell us the function of insulin. This insulin, what does it do? You tell us that now insulin will stimulate the conversion of glucose to glycogen, because glucose is too much. So insulin will stimulate the conversion of glucose to glycogen. My students, please don't say that insulin will convert glucose to glycogen. We'll get it wrong, because insulin does not convert glucose to glycogen, but insulin stimulates the conversion of glucose to glycogen. What converts this is the liver, because when you go to the university or other areas, you'll find out that we'll include here a liver. Yes, we'll include isolate of Langerhans. When you are specific to then you talk about the beta cells, the alpha cells. We will talk about those things when you go there. But for now, what is most important is you know that insulin produced from the pancreas, then that insulin is going to stimulate the conversion of glucose into glycogen. Here, that's why here I wrote that glucose is converted to glycogen. What converts it is the liver. What stimulates the liver to convert glucose to glycogen is insulin. So basically, you just know the gland, and here which hormone does it produce, and then here you come here, you talk about the function of that hormone. That is the stimulation. So basically, that's what you need to know about it. So when you go here, it's the reverse. It's the reverse. So you don't need to get stuck when you reach there. You understand? Does SAR offer lessons or videos? Yes, the lessons are there. The lessons are there. But unfortunately, I'm not a math teacher. I'm a life science and physical science teacher. So it means that I only offer here life science and physical science lessons, but maths, other subjects, all other subjects are here, but by different teachers. Yes. So if you have questions, still you can drop them in the comments. Any video you comment on, still we will bring your response, or will try to act on your what? Your question you have answered, you have asked. Anyway, let's continue. Let's go to what about when there is a decrease. If there is a decrease, yes, it means that glucose level has decreased. The last statement must be glucose level will increase. When glucose level decreases, talk about the gland. So which gland is the same? Pancreas. Pancreas is going to secrete glucagon. Don't forget that you have glucose here, glucose, and then you have glycogen. So now here you have, if you want to convert glucose to glycogen, use insulin. And then if you want to convert glycogen back to glucose, you use glucagon. Yes. So now it means that now the pancreas will secrete, because now we want more glucose because the glucose levels are low. So the pancreas is going to secrete, yes, glucagon, and then the glucagon will stimulate the liver to convert glycogen back to glucose, and then you go back to normal. Guys, this is the same situation when you have, for example, you are hungry. It's break time, you are hungry, and then after that, they don't tell you to go to maybe the teacher say that you guys you have to complete this. Then after a certain time, the hunger will go. But how did that hunger goes? It goes because now the glycogen has been converted back to glucose, and then now the body will receive this glucose and it will become normal, or it will become okay again. And then life continues. Yes. So basically, the storage form of glucose is glycogen. The storage form. In animals, the storage form of glucose, if they ask you that question, what is the storage form of glucose, is glycogen. The usable form of glycogen is glucose. The usable form of starch, that is in plants, is glucose still. So you have to know that, just so that if they bring the question, you are able to answer such questions. Now, this is the same thing I've explained. You don't need to memorize, like, no, cram. You just need to understand. First step increase, last step decrease. Which gland? Okay, you know the gland, which hormone it produces. You talked about the hormone. So what is the function of that hormone? So therefore, you have got to know the function of that hormone, and then after that, that hormone is going to bring about the decrease in the level of glucose, and then the situation goes back to normal. Glucose level in the blood increase, yes, above the normal level. The pancreas is situated in the is stimulated to secrete insulin into the blood. So what is the function of you see, it goes above normal. You talk about the gland. What does the function of, what is that gland do? It produces insulin. What is the function of the insulin? This one stimulates, you see, stimulates the liver to convert excess glucose to glycogen. You see, once excess glucose has been converted into glycogen, definitely the level of glucose will decrease and goes back to normal. That's what I was trying to explain here, but here is just in words. And then here, glucose level, decrease below the normal. So what happens? The pancreas, you talk about the gland. What's the function of the gland? Produces glucagon. What does glucagon do? The glucagon will stimulate the conversion of the stored glycogen back to glucose. The moment glucose is available in the blood, definitely glucose levels in the blood will increase, and then it goes back to normal. Simple. Simple. Guys, let me know. Is this thing simple or is that difficult? Ne? I don't think these things are difficult. You just need to put love. Some people, they call it love. Yes, put love there. Yes, the way you want it to call it. And then after that, you will see that these topics are very simple. Yes. Let's look at some of the abnormalities or the deficiencies here. So I've explained this. I've explained that. And then, yes, Young Seri is saying that you make these topics easier for me. Thank you very much, Young Seri. And I always see you whenever I do my lives. You are always there. So I credit you for that. Ne? Thank you very much. Yes. So let's continue, guys, to the next question. Yes. All right. Let's go to the next section. So here you have glucose. You have glucose. Here you have glycogen. From glucose to glycogen, you use insulin. And then from glycogen to glucose, you use glucagon. Simple. So what produces this is pancreas. And then once you know, guys, go to the paper when you know this. Ne? And then number 2, you know how to manipulate this. Simple. Once the moment you know how to manipulate this, then you will be able to know all this. So we're going to see some of the questions which are concerning about this. You'll see that the questions are simple to answer. Diabetes. Some of the conditions which you find in this glucose situations is diabetes. If the body cannot produce sufficient insulin, if the body, the pancreas, cannot produce sufficient insulin, you're going to suffer from what called diabetes. My students, please don't say diabetes. Because when you say diabetes, we will cross it. If the pancreas fails to produce sufficient insulin, insulin, then you're going to suffer from diabetes mellitus, not diabetes. Because when you say diabetes, we want to know which type of diabetes are you trying to mean? Because we have 2 types of diabetes. We have diabetes mellitus and diabetes insipidus. The diabetes of sugar and diabetes of water. So now, here, diabetes of sugar, diabetes mellitus, is divided into diabetes type 1 and diabetes type 2. Please don't say sugar diabetes. No, say diabetes mellitus. Simple. Don't complex your life. You bring in, what if I say this? What is the problem for you to memorize diabetes mellitus? Why do you want it that way? Yeah? Why do you want that way? This is what we want. Don't complex your life by losing a mark. Just say simple diabetes mellitus. Simple. You understand? Yes, you go to the paper, come out when you're happy. Don't come out of the paper when you're happy, when you're writing diabetes instead of diabetes mellitus. Let's continue. They're saying that the pancreas is not producing hormone or hormone that is insulin, meaning that insulin, you know that insulin is very important for stimulation of the conversion of glucose, the excess glucose to glycogen. So automatically, if there is insufficient insulin, it means that excess glucose will not be converted to glycogen. And then now, the body will have a lot of glucose. And then this glucose, the person will start to suffer from diabetes mellitus. How do you test for this diabetes in a simple way? Yeah, if the amount of, if you go to, you urinate, and urinate, you find glucose in your urine. That could be some of the few signs that maybe diabetes is coming. I'm not saying that if you find it, you are having it, but it could be the sign. Ne? Yes. So we are saying that diabetes type 1, what happens? The pancreas is not producing hormone, that is insulin, necessary for controlling the glucose level in the human. Diabetes type 2, that is diabetes mellitus type 2, in other words. Yes, insulin is available, but the body cell is unable to control the what? The glucose. So you find, we can bring diagrams like this, and then we ask you what is being illustrated in the above diagram. Yes, so that condition is diabetes. Here is a question. Here is a question, and the question is asking you to give us some of the answers which are here. Can I do bingo for you? Can I do bingo for you? Meaning that, can I give you a few seconds to do them, to do the questions, and then, okay. If you are on live, I'm giving you 1 minute to answer. 1 minute. 15 seconds. Yeah, 15 seconds. So that we go through and we see that what we went through or what we have gone through is correct. What you have done is correct based on what you have, what we're gonna give as the answer. Okay. 10, 8, 6, 4, 2, 0. Okay, let's go through it. Let's go through it and then we see. Okay, you're saying that you have high level of glucose in the blood. This level, yes, there is a pancreas here. Why is this organ called pancreas? Yeah, don't forget that a pancreas looks like a banana. I mean a real banana, ne? The banana for gorillas. You know, gorillas and chimpanzees like bananas. Anyway, then low levels of blood glucose, yes, but here it's still something is going to the pancreas. And then now this release of hormone 3, release of hormone 2. See? And then now you have increase blood glucose. And then here decrease blood glucose. Here comes a question. And we say, organ 1. What is organ 1? We have seen it that organ 1 is pancreas. Hormone 2. Where is hormone 2? You have high level of glucose. What do we need? We need, you see, the last high. If there is an increase in the level of glucose, the last statement must be decrease. You see what I say? Yes. Increase, it means that there is more, because here they say the high level of glucose. The last statement here is low level. Why am I comparing this? Because this is dotted, it comes like that. Yes. Meaning that now the blood glucose go back to normal. So if there is high level of glucose, we need a hormone which is going to make it decrease. So which hormone is that? Is insulin. Insulin. Meaning that insulin, hormone 2 is insulin. Definitely. Hormone 3 is what? Glucagon. Because if it is not insulin, it's glucagon. There is no any other hormone which is going to be talked about in regulation of sugars. Yes. So meaning that hormone 3 is, okay, is glucagon. They're saying that, guys, there is a confusion between glycogen and glucagon. Glucagon. Glucagon is a hormone. Glucagon is a hormone. Ne? And glycogen is a substrate, is a food. Anything which has this O becomes a food, like sucrose. Sucrose. When you say sucrase, it becomes an enzyme. So it's like a hormone, it's the same thing. You talk about, maybe you talk about lactose. lactase. So when you have A, is a hormone or enzyme. Ne? A. So glucagon. I don't know whether you read it as glucagon. I don't know. Okay, doesn't matter. Glucagon, if there is A, that's a hormone. Where is O? O means food. Don't forget that, guys. Food. O, food. Food. So you just know that this is not a hormone, this is a substrate. This is a food. Or so, guys, I think you no longer now messing it up. You're no longer messing it up. When are you going live for grade 11? Soon. I think on Friday and Saturday. I'll be coming for excretion. Yes. So we are saying that they are saying that disorder caused when gland 1 fails to release sufficient amount of hormone 2. So we don't have any disorder here. The only disorder we have is diabetes mellitus. Simple. The mechanism, when they talk about the mechanism, then you talk about negative feedback mechanism. When they talk about the condition, then you talk about homeostasis. So basically, homeostasis is a condition of the internal environment being kept constant. But what makes it to be constant is the negative feedback mechanism. I think I'm clear there. Is that difficult? You must remember. Ne? Yes. We will do more questions towards the exams. Ne? Towards the exams, as we are planning to be live for 24/7 from the beginning of prelim for all subjects until you write your end of the last question, the last paper in your final exam, meaning that you will be live for almost 2 or 3 months. Yes. So keep yourself on ThunderEDUC so that you can obtain a distinction. All the help will be here. We are saying that the mechanism that controls the level of glucose in the blood, we say that that mechanism is called negative feedback mechanism. Negative feedback mechanism. Think I'm clear there. Is there any question before I go to regulation of carbon dioxide in the body? Regulation, guys, if you have any question, let me know in the comment section. Ne? Yes, if you are looking for the distinction material book, yes, I've sent the link there. Yes, you'll find for grade 10, grade 11, and grade 12. And many books are now, we are trying to write them so that you can also benefit. Unfortunately, most of them might be out next year. But for grade 10, 11, and 12, life science, they're already there. They're uploaded. You can find them by following that link in the description below. All right, let's go to regulation of carbon dioxide in the body. Regulation of carbon dioxide in the body. How is carbon dioxide regulated? Guys, I told you that for the case of carbon dioxide, it only has one way. It doesn't have two ways. It doesn't have two ways. Let me check here. Let me check here before I go to... Okay. In this one, they brought thermoregulation. Did they? Okay, this one I'll talk about it. They bring about, no. Okay, I'll talk about, let me first describe everything, then I'll go to the questions separately. So here they're saying regulation of carbon dioxide in the body. What are you talking about here? Here, you need to know, is carbon dioxide important in our body? No. We don't need carbon dioxide. So, any level of carbon dioxide is not needed. Therefore, we have to remove out of our body. That's why carbon dioxide level in the body only has one way. It doesn't have two ways. No, it only has, if you check this one, yes, you'll find out that it has the upper one and the lower one, but carbon dioxide only has one way. Decrease in the level of carbon dioxide, that's better. If it's not there, that's good. Increase in the carbon dioxide level, we don't want it. That's why its regulation is related to decrease in the amount of carbon dioxide in the blood. Okay. First of all, carbon dioxide is supposed to be detected. When you say that, number one, someone asking me, where do I start? You start from here. I put it in number one. Stimulus. Stimulus. What's the stimulus? Increase in the carbon dioxide level in the blood. And this leads to decrease in pH and decrease in oxygen. Why is oxygen decreased? Because we use oxygen in a respiration and we produce carbon dioxide. That's why it means that if I have low level of oxygen, it means that I have high level of carbon dioxide, because oxygen has been used to produce energy, and the carbon dioxide is a byproduct of respiration. Now, how is pH decreased in the blood? How is that linked? When we look at it scientifically, sorry guys, I'm just going to use just a small bit of chemistry. Yes. We know that no one is going to ask you this, but I'm explaining to you how did this low pH come about and how is it linked to carbon dioxide. Why do we need to talk about low pH when you're talking about carbon dioxide? Yes, when carbon dioxide reacts with water, don't forget that our body is above 75% water, so we have water. Yes. So when carbon dioxide reacts with water, you form, this is a weak acid. It's a reversible equation. Okay, you form what you call carbonic acid. We call this one carbonic acid. The best example of carbonic acid is acidic rain. Rain which is acidic. Yeah, when now the first rain you're gonna receive after winter, because it has stayed longer without raining, so carbon dioxide has accumulated in the atmosphere. When that water comes and reacts with the carbon dioxide, however much is not a good, it does not dissolve in water easily, but there is some bit of dissolving, so it forms that acidic water, so acidic rain. So in this case it forms carbonic acid. So it means that now the blood, because this is water, the blood becomes a little bit of acidic. And if you have acidic acid, it means that the pH decreases, because from 1 to 6, it is acidic. So it means that the more it goes down, the more it becomes acidic, the pH becomes lower. That's why they say that increase in carbon dioxide level decreases the pH. You understand? So, meaning that whenever I have too much carbon dioxide, the pH is going to be decreased. Because our receptors in the body will not detect carbon dioxide, but will detect the pH. That's why, number 2, receptors, low pH detected. You see, how does it detect? Being detected is being detected by this carbon dioxide reacting with water to form carbonic acid. Then this carbonic acid is being detected, because carbonic acid is a chemical, is being detected by the chemoreceptors. The receptors in the body which detect these chemicals. So these chemicals are being detected by chemoreceptors. Am I clear? Am I making you lost? When is prelim section live starting? We are preparing the gadgets, yes, for helping us so that different teachers can streamline at the same time. Yes, so maybe by the end of this month, maybe around there 20 something, yes, the lives will start. Will start, so that now it is 24/7. Anytime you want to check, or if you ask a question, maybe from WhatsApp or what, we switch to it, and then, or we prepare for you, and then we answer those questions immediately. All right, let's continue to, I think I'm clear there. So number one, when you go back to what is needed in life science, increase in blood carbon dioxide levels decreases the pH or lead to the decreased pH, and then the decrease in oxygen. I think I've explained that. And then now, number two, the receptor will detect the low pH using, what are they going to use? The chemoreceptors. We call them chemoreceptors because they detect the chemicals. Yes. Number three, now the moment receptors detect, what is the next step? The receptors will detect, and then they will have to send an impulse. So these receptors, remember that receptors always the sensory cells, basically. Yes, when you go to the nervous system, the sensory cells will change this message of chemical into a nerve impulse. So impulse is sent to medulla oblongata. Why not cerebrum? Why does it go to medulla oblongata? Question. Why? Okay, it goes to medulla oblongata because medulla oblongata controls all involuntary actions, not reflex actions. There is a difference between involuntary actions and reflex actions. Actions which are done to protect the body from danger, we call them what? We call them reflex actions. But actions which work without control, but they are not linked to protection of the body, then we call them involuntary actions. For example, breathing, heartbeat, peristalsis. When you are swallowing the food, when food goes in the throat, you can't tell the food that, come back my food. No way, it has gone. The sweetness is gone. Once it reaches the throat, down the stomach. You can't control. You are sleeping as the food is moving. How did it go to the rectum? So that you can go to the toilet to do the best action ever. Yeah. How did it go there? It goes there. You don't have control over that. All those actions are being controlled by medulla oblongata. Hence, the impulse is being sent to medulla oblongata. So what does the medulla oblongata do? The medulla oblongata will send a message, another impulse, to respiratory muscles. That's the diaphragm and the intercostal muscles. Yes. To increase the rate and the depth of breathing. Heartbeat. Meaning that the heartbeat will increase. It does not only increase the heartbeat, but even the depth. The rate, that is fast, the rate, depth. You understand? The depth increases and the rate at which you are breathing also increases. Why does it increase? Why do we need to increase the breathing rate? And the heart is supposed to increase the rate at which it is pumping blood, so that this blood can transport this carbon dioxide from the cells where carbon dioxide is being produced from to respiratory surfaces or respiratory centers, that is the lungs. And more oxygen is taken from the lungs to the muscles or to the cells where this oxygen is needed, so that now the level of carbon dioxide decrease and the level of oxygen increase in the blood. You understand? This thing is working antagonistically, because here we started with carbon dioxide increase and oxygen decrease. So what we are looking at, carbon dioxide to decrease and the oxygen to increase. Therefore, the heartbeat increase. Don't forget that the heartbeat originates from within itself. It has muscles. Yes, tissues. There are muscles, tissues, which initiate the heartbeat. But here, this medulla oblongata, what it does, it increases the rate at which the heart is beating. Yes, when you go to the heart, you will study about it, that it has its beating. The beating is originated within itself. And always I tell my students, we don't determine that the person is dead by using the heart. No. You can remove the heart, hold it, and the person is still alive. What about those people where heart transplant is being done? The hearts are being removed. Yes. So death is not related to the heart. Why? Because the heart has its own beating mechanism. Death is regarded to the brain. If the brain dies, then automatically you are dead. However much the heartbeat is still there, you are dead. Yes. So the heart beat, this medulla oblongata. Why am I saying that? I'm just trying to show you that the function of medulla oblongata is not to start the heartbeat. No, to increase the rate of heartbeat so that more carbon dioxide is removed from the body. And the moment carbon dioxide is removed from the body, definitely the carbon dioxide level is going to fall down. What about the pH? The pH is going to increase back. It's going to go back to normal. Why? Because now there is no carbon dioxide reacting to this, or if it is there, it's very minimal, reacting with water to form carbonic acid. It means that less carbonic acid is being formed. And definitely what happens? The level of carbon dioxide goes back to normal. And that's the mechanism. I don't know why they like this question so much. Maybe because students tended to fail this question, but they like to ask this question. And this is the only question, can I repeat? Is the only question in the negative feedback mechanism where it is brought as it is that describe the negative feedback mechanism of carbon dioxide. The other questions of this homeostasis negative feedback mechanism, they only come in different versions the way I'm going to show you. But this is the only question which comes straight. And in most cases, when it comes for six marks. So guys, just in case it comes, make sure that you go when you know it. It must not be an issue for you to fail. Yes. Let me know if you have any question. Yes, so that I can also smash it before I continue. So let me do a wind up. Number one, stimulus. What is the stimulus? Increase in carbon dioxide level. This leads to the decrease in the pH and decrease the level of oxygen. I explained it, that oxygen decreases because it's being used under respiration and carbon dioxide is being produced, and carbon dioxide reacts with water to form a low pH. And then now the pH is being detected. Because the pH is low, is being detected by the chemoreceptors. Why chemoreceptors? Because chemoreceptors, they detect chemicals, and low pH carbonic acid is a chemical. So now this will, the control centers will send the impulse to medulla oblongata. Why medulla oblongata? We say that it's medulla oblongata because medulla oblongata controls all involuntary actions. The respiratory muscles of the diaphragm and the intercostal muscles, the increase in the rate and the depth of breathing, and muscles of the heart increase. So the moment all this increased breathing increases with the depth and the rate of heartbeat also increases, definitely more carbon dioxide is going to be removed from the body. And then if carbon dioxide is removed from the body, then automatically the pH is going to return to normal. And then it continues like that. I think I'm clear there. And I've said that this is the only question where they bring it as the way it is. You write it the way it is. You don't draw flow diagram. No, please don't draw flow diagrams. We won't give you a mark. write step 1 in paragraphs. You can write in paragraphs or in bullets. Still is fine. Still you get the mark. Yes. Are we together? Are we together, guys? Yeah, so basically this is an explanation in words of what I've explained. How to write in exam: high concentration of carbon dioxide lead to formation of carbonic acid. The pH of the blood drops, meaning that there is more acidic. It's more acidic. The impulse is sent to medulla oblongata, and the breathing rate and the depth of breathing increase. The heart rate also increase, and more carbon dioxide move to the lungs. Yes, so that it is exhaled. Yes, breathe out. All those words can be used concurrently. They're saying that then the blood carbon dioxide level return to normal. Simple. Hey, that's my slogan. I like it because these things are simple, but sometimes the books make them to be difficult. Use this distinction material. There is no way how you can fail to get a distinction. Yeah, download it from the description below. Yes. So, it's simple. It's simple. I always tell my students that it's simple. Things are simple as if you are taking ice cream, because there are no bonds. Anyway, let's look at regulation of water. Yes, we're going to look at regulation of water, regulation of salt, then thermoregulation. Yes. After that, maybe we shall use some 20 minutes and then we discuss about the questions you might find in the paper. If I don't exhaust all of them, definitely I'll come back again. Only for questions. Only for questions. Yes. So that at least, and I think I should make it either Microsoft or Zoom or website, any of those options. Yeah, meaning that if I don't make it live on YouTube, then automatically I'll make it live the other way. Yes. Okay, so we are done with the carbon dioxide. Let's go to salt. These ones are simple. These ones are simple. Let me show you that how simple they are. Regulation of water, of water balance in the internal environment, which we call osmoregulation. Whenever you talk about osmo, we mean whenever you talk about osmo means water. Don't forget that we said osmosis, movement of water molecules from region of low concentration to region of high concentration across a semi-permeable membrane, or movement of water molecules from a region of their high concentration. The water molecules where they are highly concentrated, meaning that it's a dilute solution, to a region of what? They are low concentration, meaning that a high concentrated solution across a semi-permeable membrane. Meaning that whenever I talk about osmo, we are talking about water. So, don't forget the technique I taught you, that if you have decrease, here is decrease, it starts with decrease. By the way, guys, it does not mean that because this thing is facing up, then it must be always increase. No, it is not up. This is not up, and this is not down. It's just a screen. It's just a screen. When you draw, when you do Africa, and you see that the river is flowing like that. Have you ever seen a river flowing from low to high? No, there is no river. It's just a paper, or it's just a screen. So this is not necessarily meaning that this is up, this is down. No. So please take note of that. So it must not confuse you that whenever you see something going up, then it is up. No. So now we are saying that there is a decrease, definitely the last more, it must be increase or rise or more. Anything which is related to increase. You understand? It must be, because we started with the decrease. So now the 2 steps are done. Now I need to know the gland, which hormone does it produce, and the function of the hormone being produced here. And then I'm done. The negative feedback mechanism or the osmoregulation is done. Simple. Okay. Water level decrease. The pituitary gland. Some students, they have confusion here. What hormone produces ADH? Sorry, which gland produces ADH? The gland is hypothalamus. Why are we talking about pituitary gland here? Okay, let me give you an example, an answer. The hormone ADH, antidiuretic hormone, make sure that you know how to write it. Antidiuretic hormone, yes, is being produced by hypothalamus, but is stored in the pituitary gland. So when you use hypothalamus here, make sure that you know how to link it. So is stored because the pituitary gland or hypophysis, sometimes you call it hypophysis. Please don't call it master gland. Please, we won't give you a tick. If I ask you what is my name and you tell me I'm teacher, that's not my name. That's my job. So when you call it master gland because it controls most of the glands. So please don't call it master gland. Call it hypophysis or pituitary gland. Its name is pituitary gland. You understand? Or hypophysis is cool. So secrete more ADH. What is the function of ADH and where does it work? ADH works in the kidney. It works in the kidney where the nephrons are. Yes. So it works in the kidney where in the nephron. It brings about high permeability. Permeable, when you talk about permeability, we are talking about allowing some substances to go through something. Yes. So we call it permeability. Highly permeable. You understand? We call it highly permeable. So it allows, when you talk about high permeability, allows some substances to go through. So ADH, the function of ADH, it goes to the nephron. I think you remember the structure of a nephron. If let me show you here. In the distal convoluted tubule, because this is grade 11 work, but some people now, they have forgotten the structure of a nephron, but it's simple. Let me show you how it looks like. Okay, if this is the Bowman's capsule, it's graded. I'm just drawing a simple thing so that you can understand it better. This is the proximal convoluted tubule. Yes, it goes down like that. Proximal convoluted tubule. Yes, now you have the descending loop of Henle, and then you have the ascending loop of Henle. Then you have yes, you have the distal convoluted tubule. The distal convoluted tubule, and then it's connected to the collecting duct. And here you have the afferent vessel. Here you have the glomerulus. Yes, the glomerulus, and then you have the efferent vessel. Afferent vessel must be bigger than the efferent vessel. That's not a big deal. That's grade 11 one. So what I want is ADH. This ADH will come here in the distal convoluted tubule and the collecting duct. This is the collecting duct which connects to the ureter and then to the bladder. Yes, it comes here and makes it more permeable. Because we have less amount of water in the blood, meaning that you're urinating urine which is yellowish. When you urinate urine which is yellowish, you just know that you have less amount of water in the blood. Therefore, you're supposed to drink more water, meaning that more water is being reabsorbed back to the blood. Or if you eat something which is salty, if you eat meat, people who go to cheese and nyama, yes, you eat that hot, nyama, yes, please make sure that you drink water. Why? Because these proteins, they require more water for it to be metabolized. Yes. So now what happens is this ADH will make this nephron, the walls of the nephron, this one, the distal convoluted tubule, to become more permeable. When it becomes more permeable, it means that now more blood, more water is being reabsorbed. The word is reabsorbed back to the blood. And then now less is being lost as urine, because what remains here in this tube is urine. So meaning that less water is lost through urine, and then now more water is being reabsorbed back to the nephron, to the bloody vessels. So that's why here we are saying that the pituitary gland will secrete ADH. That one is fine, but this ADH must be more. What is the function of ADH? I explained this so that you understand the idea behind. The permeability of the collecting duct and the distal convoluted tubules will increase. The permeability, meaning that it will become more permeable. It will allow more water to go through. You understand? Yes. So where here in the distal convoluted tubule and the collecting duct, and then if more water is being reabsorbed back to the blood, meaning that more water is the word is guys. Please don't use more is absorbed. We will cross it with the happiness. Why happiness? Because I've told you and you have not taken it as a note. Please don't use absorption. Is reabsorption. Now someone can ask me, why reabsorption? Is because the first absorption occurs in the digestive system. This is the second absorption. Hence we say reabsorption of water to the blood. Can I please have this video after the lesson? Yes, the video will be there. Yes. Guys, don't forget to subscribe, like, and share to others. Yes. So we are saying that I think I've answered you. They're saying that more water is reabsorbed back to blood. And then once more water is reabsorbed, automatically the water level increase. That's why you say that decrease here. The last statements must be increased. More water is reabsorbed back to the blood. In other words, you're gonna produce more concentrated urine. The urine is gonna be more yellowish. Less water is gonna be lost as urine, and more salts are gonna be lost as urine. That's the meaning. What about when the level of water in the blood is too much? We said that our body is made up of over 75% water. I hope we need water. Don't forget that too much of anything is always bad. Meaning that however much water is important in our life, but too much of it is bad. Yes. So, water level increase. Definitely the last statement must be water level will decrease, or anything which is related to decrease. Less water, more water. I think. So now I need to know which gland. I know the gland, meaning the pituitary gland. So what's the function of pituitary gland? Is gonna, because I have more water, I don't need more reabsorption here, then automatically I'm gonna produce less ADH, or completely stops. ADH will completely stop depending on how much water is there or is needed in the blood. And then now, meaning that the pituitary gland, the hypophysis, is going to stop secreting water. Yes, the water, the permeability of the collecting duct and the distal convoluted tubule will decrease, meaning that less water is going to be reabsorbed, meaning that more of it is going to be lost as urine. This is the time when you urinate urine which is yellowish, colorless, not yellowish. Colorless, meaning that you have a lot of water in the blood. And this is the time where I don't think you might feel thirsty. You might have that thirstiness. Why? Because you have a lot of water in the blood. This is it. When water level is decreased, you produce yellowish urine. That's the time where you're gonna be thirsty. Yes. And if water is colorless, then that's the time when the water is not going to be, the urine is not going to be thirsty. Did I say thirsty? Colorless. So when it is yellowish, smelling too much because of the other waste products which are there. But when it's like this, then automatically, when it's colorless, it's not going to be that you are smelling, because the water has diluted those other ions and wastes. So basically, when there is too much water, the last statement must be decreased. The pituitary gland will stop or produce less ADH. You use 1. You don't write / yes. The permeability of the collecting duct and the distal convoluted tubule will decrease, and then less water is going to be reabsorbed back to blood. And meaning that more is going to be lost as urine. Guys, am I clear? Are you getting it? Do you have anything you have added to your knowledge today? Yes? Do you have anything? Young Sarah is asking, when is the next class? Friday and Saturday. Yes, Friday and Saturdays. All right. Let's continue and say that when blood has more water, okay, this is just a description of what we have explained. When a blood has less water than the normal, the hypothalamus is stimulated. You see, I told you that you need to know how to connect if you are using the word hypothalamus or you are using the pituitary gland. Both of them are fine as long as you explain them correctly. Then the hypothalamus is going to be stimulated and is going to send the impulse to the pituitary gland. Why it sends the impulse to the pituitary gland? Because the pituitary gland stores it. And then now the pituitary gland is going to secrete more ADH. You see? Then this ADH is going to travel in the blood to the kidney. Where exactly in the kidney? In the nephron. And then this ADH is going to increase. What's the function of that hormone? Is to increase the permeability of the nephron. And then after that, water is reabsorbed back to the blood. Meaning that less is lost as what? As urine. So water level in the blood returns back to normal. And then the other way is the same thing. When blood has more water than the normal, the hypothalamus is stimulated, and then the impulse is sent to the pituitary gland. And then what happens? Less ADH, or no ADH is produced. So you choose whatever you want to write, either less or no, or depending on the scenario they have brought you in the exam. So less travel in the blood to the kidney. And then what happens? There is decrease in the permeability, meaning that the walls, they're going to become less permeable. It does not allow water to go through. And then now less water is reabsorbed back to blood, meaning that more is lost as what? As urine. Here less is lost as urine. Here more is lost as urine. This is yellowish urine, and then this is colorless urine. Yes. And then now this is smelling urine, and this is, I don't know, odorless. Water level in the blood returns back to normal. I think the way how I've explained it here. What about the salts? So you guys, we are remaining with the salts and thermoregulation. I'll show you how you can go to the paper without stress and you pass everything. Why not passing? You have to. Yes. Because they set these things so that you guys, you can pass. So you guys make sure that you pass. Make sure that you pass. And here they're saying that regulation of salt balance. How to write in exam. But before you know how to write in exam, you need to know how to work upon it. So it's the same thing, guys. It's the same, exactly the same thing. If you are saying that normal level, cool. Increase here, decrease in the level of salt, yes. The last statement will be increase in the level of salt. We know that. Then you need to know the gland which is produced, which is going to act upon the salt, is adrenal gland. Don't forget that adrenal gland produces two hormones. The first hormone is aldosterone. The second one is adrenaline. So in this case, we don't want adrenaline because adrenaline is used as emergency. It's a fight or flight. You either take off, or you have to fight. So that is adrenaline. We're not interested in adrenaline now. We're interested in aldosterone. So aldosterone is going to be produced because we have less. So what's the function of aldosterone? It also brings about the permeability. It brings about more reabsorption. I said that always use the word reabsorption. So adrenal gland, what's happening with adrenal gland? Adrenal gland is going to produce more aldosterone, and this aldosterone is going to make the nephron to reabsorb more sodium ions. The moment sodium ions are reabsorbed, the salt level will increase and goes back to normal. What about when the salt levels are increased in the blood? Then the last statement must be decreased. So adrenal gland will stop or will secrete less aldosterone, one of the two. And then now it means that it will be less reabsorption of sodium ions, or there is decrease. The sodium ions reabsorption will decrease, and the moment decrease, the level of salts will go back to normal. Now, this salt, aldosterone, aldo, which means salt, salt, aldo, just for you to remember. It's just for you to remember. It's not a big deal. Yes, this aldosterone works together with ADH. If there is too much salt, sodium ions, too much salt, yes, meaning that there will be less reabsorption of salt. It means that there is less water. Too much salt, it means less water. Too much water, if there is too much water, then it means that there will be less salt. So these two, they work antagonistic in the opposite way. Yes. So meaning that when ADH brings about more reabsorption, there is a question I saw. I'm gonna give it to you so that I show you how you can answer, how you can bring about the interaction of aldosterone and the ADH at the same time. So that you must know that when too much water is there, you urinate urine which is colorless, it means that urine has less salts but too much water. When you urinate urine which is yellowish, it means that that urine has more salts but less water. And that's exactly what happens in the body. Yes. So I think I'm clear there. Then how do we write in exam? How do we write in exam? We write in exam that when the salt level in the blood increases, yes, the adrenal gland will be stimulated. Will be stimulated. When it's stimulated, what happens? It's gonna secrete less. If it is less, it's gonna produce less. It's gonna produce what? Less aldosterone. If less aldosterone is there, it means that there's gonna be less reabsorption because there is too much salt. Yes, more less reabsorption, and if less reabsorption occurs, what happens? It means that there will be a decrease. There will be a decrease in the the salt concentration in the blood, and then to turn back to normal. What about when there is increase in the salt? So there is decrease in the salt. So the last statement must be increase, as I told you, meaning that the adrenal gland will become more stimulated, meaning that more aldosterone is going to be produced. And if more produced, meaning that there will be more reabsorption. That's why you're seeing that increase in the reabsorption of sodium ions. And then once more sodium ions are reabsorbed back to the blood, meaning that the level of salts in the blood will increase and it will turn back to normal. So you see that things are not as that difficult as you see them. The only challenge which happens is when it comes to the exam, sometimes, my students, you go off. So after covering this, then it means that I'm going to be focusing more on these topics. You understand? So the next topic after this, a link so that at least you don't get lost in that regard. Let's go to the last subsection. What is that? Is the process of thermoregulation. The process of thermoregulation. Here, when you talk about thermoregulation, what comes into your mind? Thermo means heat, temperature, thermometer. Whenever I talk about thermo, we have to talk about heat, temperature. Regulation is control. Basically, you are controlling the heat, the amount of heat in the body. So when you talk about thermoregulation, is controlling the amount of the heat in the body. Remember that we are endotherms. We're supposed to keep our internal environment constant. That's why thermoregulation is also an example of homeostasis. It's supposed to be kept constant. Above it is problem. Below it is problem. Meaning that we're supposed to regulate at 35.5 degrees Celsius. This is 5, not 8. Ne? Yeah, 35.5 degrees Celsius. Yes, at least there. 35 to 30. Yeah. Did I say 35? 37. 37.5 degrees Celsius. So above that, yes, above, not 30, 35, ne? 37. I was talking about the temperature at which sperms are being produced. Anyway, it's 37.5 degrees Celsius. So above it is problem. Below it is what? A problem. Meaning that when it's above it, it will be denaturation of enzymes. And then you won't work. You won't have hormones. You won't have enzymes. Because all those things are protein in nature. Anything which is protein in nature will be denatured. And then below it, it will become inactive. inactive, meaning that it will be slow, it will be dormant. Yes, like the same way some creatures are dormant in winter because the temperatures are low. What happens when it's too hot? You go to the houses also, meaning that too much of the temperature is bad. Too low of temperature is bad. Anything which is too much is always bad. Anyway, let's go back to what brought us here. So, we're saying that thermoregulation is the process that allows your body to maintain its core internal temperature. All thermoregulation mechanisms are designated or designed to return your body temperature to a steady one. So the body temperature is regulated by hypothalamus in the brain, meaning that the hypothalamus is supposed to communicate to the blood vessels and the sweat gland, meaning that during this process we're going to talk about more of hypothalamus, the sweat gland, and the blood vessels of the skin. So once you know that, that's when you talk about the thermoregulation, you must know that that's what you are supposed to talk about. Please don't forget this, because this is your future. Okay, before you go there, you need to know something very small. If I bring a diagram for you, can you identify for me the following? Number one, the things which you need to know is sweat gland, sweat duct, sweat pore, sweat. Yes. Sweat gland, sweat duct, sweat pore, sweat. Then the blood capillaries. So when you talk about sweat gland, yes, when you talk about sweat gland, we talk about this. Sweat gland. Yes. This is the sweat gland we are talking about. Sweat gland. Sweat duct, a tube which transports the sweat. Sweat duct. Sweat duct is this one. And then sweat pore is the hole where sweat moves out. Is this one. Sweat pore. Where you see your liquid when you are too vigorous with an activity. Then the liquid which comes out. So you have to know how to label it. Then number two, you need to know the blood capillaries. The blood capillaries. This is a hair follicle. So the blood capillaries, you must know them, how they work. Yes. So now let's come here. So this is the blood capillaries, and this is the sweat I was talking about. So sweat gland, sweat duct, it's a sweat duct. Then this is a sweat pore, and then this is sweat. So you need to know that. And then these are the blood vessels, blood capillaries. So we have this shunt. During hot day, it shrinks and more blood will pass through near the skin. And then during cold day, then this opens and then this shrinks, and then more blood, this becomes bigger. Let me show you. It becomes bigger, you see, and then this one shrinks, and then more blood will be passing down the skin. And that's why during hot day, you cannot see, during cold day, you cannot see blood vessels on your skin easily. You can see, but you cannot see them easily the way how it is hot. Yes. So now, what happens during this thermoregulation? What happens? Let's link now. Let's link to the question.
All right. Let's go back to the question and then we see what happens on hot day. So now we have to discuss about how do we bring questions concerning about this. Yes. Anyway, let's go. The first statement you need to talk about is the hypothalamus, because it is the hypothalamus which detects the change in the temperature. Don't forget that we said that the function of hypothalamus, when you go back here to this, the hypothalamus, yes, we say that is responsible for the hormone it produces is ADH. But here we say that yes, we talked about it when you're talking about the hormones. Down here on this booklet, let me show you, just to remind you something small, here. Guys, make sure that you have this distinction material so that you can, yes, hypothalamus, where are you? Say, I'm here. Okay, it's here. All right. Control center for hunger, thirsty, sleep, body temperature. That's why you talk about hypothalamus. Because hypothalamus is responsible for body temperature and emotions. Are you always happy, sad, bipolar? Meaning that now you're happy, next time you are sad. What controls that is hypothalamus. Anyway, this is not today's subtopic. Today's subtopic is just to know why do we talk about a hypothalamus? Because we said here, hypothalamus is the center for temperature. Now, let's go back to our description. Yes. So, now when you're saying that, hypothalamus is stimulated. Now, I think you know the reason why it's stimulated. During hot day, when you talk about hot day, hypothalamus is stimulated. When it is stimulated, what happens next? Yes, it sends the impulse to the blood vessels. Meaning that these blood vessels, this hypothalamus is going to send the impulse to these blood vessels. What will happen to the blood vessels? The blood vessels will dilate. They will become big. The blood vessels of the skin, meaning that they will become more wider. This is called vasodilation. Guys, listen carefully. You're saying that hypothalamus is stimulated and sends the impulse to the blood vessels. That's the mark. The blood vessels will dilate. That's the mark. This is called vasodilation. Is another mark. So don't write the blood vessels will become dilated and you skip. You go to more blood, you will float near the skin. No, this is called vasodilation. We give you a tick for that. This is called vasodilation. More blood flows to the skin, meaning that we will experience more blood flowing near the skin. Yes. And then more heart. Yes. Here you have more heat is being lost from the skin. If more blood is flowing near the skin, meaning that you're going to have a lot of heat being lost. That's why you see these arrows there. Why? Because this blood is hot. So if it's hot, meaning that more blood is going to be what? It's going to be lost. More blood is going to be what? It's going to be lost. And then, if more blood is being lost, what happens? It means that, more heat is going to be lost. What happens? Meaning that it's going to make the blood to be cooled a bit. But it does not stop there. More blood is sent to the sweat gland, meaning that now this blood is going to go to the sweat gland. More blood is going to be sent to the sweat gland. Now, the moment more blood is sent to the sweat gland, what happens when you put too much food in the mouth? You just stop chewing, or you increase the rate at which you are chewing. You increase. So if more blood is sent to the sweat gland, it means that the sweat gland will become more active. The sweat gland is going to become more active. And once it becomes more active, it means that more sweat now will build. You understand? And then now you'll see your skin like this.
meaning that more sweat is being produced, is being released. So the sweat gland will become more active, meaning that it's going to produce more sweat. More sweat is being released, and once it releases, is being released, it means that now, as it's being released, evaporation is going to take place. And evaporation to take place, you require heat to evaporate this water. And as evaporation is taking place, it leads the skin to be cooled. It leads to the cooling of the body. So evaporation of the sweat cools the skin. That's the meaning. Yes, that's the meaning. So that evaporation leads to the cooling of the skin. Sebastian, brilliant. Okay, thank you very much. Yeah, actually, you are more brilliant, the one who is listening. Yeah, than me who is talking, because when you are taking the information into the head. Guys, let me know if you have a question, so that we sort that out. If you have any challenge, don't be scared to ask a question. Don't be scared. Ask it, we smash it, so that at least you don't lose it. Yeah, you don't lose it. So basically, that's what happens during hot day. What happens during cold day? What happens? So the hypothalamus, because it's the same gland, is going to be stimulated. But in this case, when it's stimulated, it has to send something. The impulse sent must be just a little different. Okay, the impulse is going to be sent to the blood vessels. The blood vessels will constrict. Guys, please, those who are having the distinction material need to rectify this. Yes. The blood vessels become wider. No. The blood vessels will become narrow or small. Yes. They'll become narrower. Yes. This is called vasoconstriction. The blood vessel of the skin will become narrow. This is called vasoconstriction. Check. Check. The upper side is narrower. The lower side is big, wider. This is a tick, a tick, a tick. Yes. Guys, did you know that it is nice to mark a student when you are just giving it tick, 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 tick? Make the hand of the examiner just to tick, so that even if he's wrong, they just give you tick, 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 tick. Then when you get wrong, the examiner starts to say wrong, even the correct one wrong. Aibo. Make the examiner to be impressed that everything you are writing is correct. Yeah, how are you going to make the examiner? Use this distinction material. Download it, guys, and get your copy. Watch these videos. ThunderEDUC. Yeah, you always. There is no way. I remember this guy who WhatsApped me and said that, sir, I got 12 distinctions from my class. He was a teacher. They said, this is brilliant. 12 distinctions. He had never got a distinction before. But when he started using this distinction material, distinctions now. Distinctions are just flowing like water in the river. Flowing water in the river. Water in the river. Water in the river. Yeah, so make sure that use this distinction material. You will have distinctions flowing like water flowing in the what? In the river. Yes. So we're saying that if this is vasoconstriction, less blood will flow near the skin. We have explained it. Less heat is gonna be lost because less blood is being is flowing near the skin. It's like you when you are sleeping without a blanket, more heat is lost. What about when you have a blanket? Meaning that the heat will take much more time to cross this blanket to go out. Meaning that less heat is gonna be lost. Yes. So less blood is sent to the sweat gland. If less blood is sent to the sweat gland, meaning that the sweat gland is going to become less active. What happens if you put less food in the mouth? Definitely, you'll chew less. That's the same thing. Meaning that here, it's not that much, meaning that you see less sweat is being produced. Meaning that you won't have that much sweat. Like in winter. Yes, less sweat is released and then less evaporation. And if less evaporation is happening, it means that there will be less cooling of the body. Then you'll be like this. Why do you have these goosebumps? And the hair are going up. Anyway, this hair rising is also another mechanism of keeping internal environment constant. Unfortunately, we don't talk about it in this case. But I would have explained it to you. If you go to the university, they will explain it better when you are using this erector peel muscle, which contract and make this hair to stand upright. Yes, anyway, you're not talking about it. Let me not put you in the stress. Wow, here are some of the questions. Yes, here are some of the questions which you need to answer for me. Can I give you 2 minutes to answer for me how to explain the negative feedback mechanism of carbon dioxide when a person is running faster? I explained it. Young Sir, I explained. It's simple thing. The way. That's why I told you that the carbon dioxide. Wait, guys who are starting to write. The carbon dioxide is the same. Is the same. You don't need to have stress with it. That's exactly what you need to write. Yes. So meaning that when the person is running faster, it means that more carbon dioxide, more oxygen is being used, meaning that respiration is taking place, meaning that more carbon dioxide is being produced. Meaning if there is more carbon dioxide being produced, meaning that there will be more carbonic acid which is being formed, and the pH of the blood will drop. You understand? And then the impulse is going to be sent to the medulla oblongata, and then the breathing rate and the depth of the breathing is going to increase. You understand? And it doesn't stop there. The heart rate also increases. And then now more carbon dioxide will be sent to the lungs through the blood. And then the lungs is going to exhale this carbon dioxide from the blood. And then the level of carbon dioxide will return back to normal. And once it returns back to normal, then the person will go back to normal in terms of the heartbeat and the breathing. Exactly. I said that negative feedback mechanism for carbon dioxide is the only mechanism where whatever case they bring it, you're going to have to write that. You understand? Yes. You're going to have to write that. And then it doesn't have the other way around, meaning that there is no way how you're going to say that, okay, the top side or the bottom side. Which one am I going to choose? You only choose one. That is it. So carbon dioxide, whatever case they bring it, you write the same thing. Make sure that you memorize it and then you get it right. I think, Young Seri, I think I've tried to explain that. All right, let's go through this. And then we say that. Identify the part labeled A. Where is A? A, yes, we have said it, that it is sweat gland. Yes. Give the number of the diagram 1, 2, 3. 1. Oh, give the number of diagram 1, 2, 3 that represent the body's response to high environmental temperatures. Here we have to look at what do I look at if the temperature is high? If the temperature is high, yes, if the temperature is high, you need to look at the blood vessels. They must be dilated, and you need to look at the sweat being produced. You can't look at the sweat gland because the sweat gland, you don't know whether it's more active or not active. The only thing you know that is active or not active by looking at the amount of sweat which is being produced. So on the hot day, it's going to be this and this. Because here they check the blood vessels. These ones, they are more wider compared to this. You see, they are more wider. Check this size. They are more wider compared to this. They're saying that would the skin release more heat through radiation? Diagram 1 on diagram 3. Remember, radiation is a loss of heat without contact, meaning that like the sun, the way how it brings heat to us, you don't touch the sun. Yes. So they're saying that would the skin release more heat through radiation in diagram 1 or 3? Diagram 1 or 3. Because this one is under high temperature, then it's going to lose it diagram 3. Give number of the diagram 1, 2, 3 that represent vasoconstriction. 1, 2, 3. Vasoconstriction is diagram 1. Why? Because the blood vessels are constricted, meaning that they are small, they are narrow. They're saying that which part of the brain controls thermoregulation? Hypothalamus. We have talked about it, and then even I showed you about it. What about this question? They're saying the diagram below represents the structure of the skin. Structure of the skin. They are saying that if this is the structure of the skin of two people, both people were in the same room at the same time, but one person was exercising while another one was not exercising, meaning that he was sitting still. The skin surface temperature of both people was measured after 10 minutes. Okay. Which person A or B was exercising? Let's look at what we said. Here there is sweat, and the blood vessels here are more wider, meaning there is vasodilation. So it's this one who was exercising. Give two visible reasons: production of sweat, and then the blood vessels are more wider. Give one hormone that would have the same effect on the blood vessels that is observed in A. In A, there's vasoconstriction. You see? If it is vasoconstriction, then definitely the hormone is going to be adrenaline. Why? Because during emergence, we said that the blood vessels of the skin constrict. Let me show you. This distinction material has this answer. As I told you that there is no way how you're gonna be asked a question and the distinction material, this book cannot answer it. So let me show you where it is. You go to where am I here? Where am I? Yes. So it's around here. Yes. Let me link what is this? Okay. Okay. Now it's around here. Sympathetic and parasympathetic. Yes. Check. We say that sympathetic nervous system work together with adrenaline. You see? And then what happens? During the sympathetic nervous system, that's when adrenaline works together with the sympathetic nervous system to bring about emergence. There will be increase in the heart rate. Cool. Constriction of blood vessels in the skin, vasoconstriction. You see? Definitely, this is what they are trying to say in that question. Wabon. So, if we go back to add that question, yes, if you go back to this question, here, they're saying that name one hormone that would have the same effect as blood vessels that is observable in the person 1. This is vasoconstriction, and so the hormone is gonna be adrenaline. Yes. After 10 minutes, the surface skin temperature of each person was measured. Okay, if the surface skin temperature was measured, so what does it mean? It means that the person A, so explain why the skin temperature of person A was higher after 10 minutes. And person A was the one who was exercising. You see? So why is nope. Explain why the skin temperature of person let's find out. Did they say person A is the one who was exercising? They're saying after 10 minutes of the skin surface, okay, the person was measured. The result is here. So they're saying that explain why the skin temperature of person A was higher after. Where is person A? We need to find out. Person A. They're saying the diagram below represents both people, okay, exercising, and another person was sitting still. The skin surface temperature of the was measured after 10 minutes, meaning that this one was exercising while this one was not exercising, because of what we are seeing here, because of the heat. And even we said it, I think somewhere here, who was exercising, and we say that person B was the one who was exercising, meaning that person A, his temperature was higher. Person A, temperature was higher, but this person was not exercising. While person B, the skin temperature was lower, and this person was exercising. But this only happens after 10 minutes. What caused this? It's because of this. It's because of this. Meaning that more heat was lost to the surrounding. The moment more heat was lost, the surrounding was being absorbed by, and then now this liquid, the heat, the sweat, also led to the cooling. And then the moment it cools, so when the air blows, on this one becomes colder. You understand? And then now the skin surface, it becomes much more colder. It's like when you take a warm bath. Yes, you bath. When you bath, it is you are warm. But when you remove out of the warm bath, yes, and then you expose your skin to cold air, you become more colder than before. That's exactly what happens here. That this person, because the person A was not exercising, so was not exposed to any coldness, any liquid which is going to make the skin to be more cold. But now, because this one was exercising, and then the sweat was there, and then the sweat made the skin surface to lose the heat, then the skin surface became more colder. That's why this person is having a lesser temperature compared to this, but only at the skin. There is another question. There are so many questions, guys. Yes. Here is a question. They're saying that normal healthy person was placed on. Okay. If there is any question, please let me know. Yes. They are saying that a normal healthy person was placed in a cold room after 30 minutes. The thermal image shown below was placed. Okay. This thermal image indicates the temperature of different parts of the body. Lighter colors of the scan indicates the temperature lower than the normal temperature. Oh, okay. Meaning that this is lower than this is lower than this. Which color, gray, black, or white, represents the body temperature? Definitely is black. Why? Because they say these are lower than. So this is the normal body temperature. So the answer becomes black. State what would state what occurred in each of the following parts in the person's skin during the temperature regulation. Okay, blood vessels. Now you need to find out, was it in a hot environment or in a cold environment? Cold room. Definitely, you're gonna have, if it is cold room, then automatically blood vessels will go constrict. Yes, this is what I'm saying. They will constrict. This is what I'm saying. Because it is on a cold day, constrict, vasoconstriction. They'll become more narrower. And then sweat gland. The sweat gland will become less active. Less active. The sweat gland will become less active. That's how. So it's going to become less active. And they're saying, name the part of the brain that is responsible for thermoregulation. Hypothalamus. We have said it several times. Hypothalamus. Yes, there is no way, guys, if you follow this distinction material, you're gonna fail. Name the part of the brain, okay? State one way in which the thermal image would differ if the person was placed in the hot room for 30 minutes. So how will it differ? Yes, how will this? Definitely, automatically, it means that these light colors will become much more darker because the heat is being absorbed from the surrounding.